Thank you, Prof. Welcome to everybody, and thank you once again for joining us. I'm just going to do a brief recap on some of the aspects today, just as a refresher, and then we'll move on to today's session, which will basically cover, some of the basic aspects of NMR as well as the applications thereof. So I think some of the questions that were raised yesterday pertain to the use of plastics. And I think Prof emphasized that for the amount of time in which the sample is in that particular container, it really doesn't lead to any of the plastic in your sample. I also posted that at the NWU specifically, at our campus, what we use is the low bind Eppendorf tubes. So there were some tests done in our lab also that showed that there was minimal leaching of, the plastic into our samples. And this was done with GC, with gas chromatography. So one of our GC systems. Then there was also a question regarding bacteria. And I think Prof emphasized that you can run, a range of samples on NMR. The key thing is that you basically detect then the proton or the hydrogen. Prof Prinsloo showed you guys a video on the apolar as well as polar extraction of metabolites from plant material. And I think there was, for the apolar sample, Prof mentioned that the pH stays more or less constant. It doesn't vary as much with the apolar extraction, and therefore there was no adjustment needed of that. In terms of how pH is maintained, with respect to, more of the human or biological side, I'm just going to ask Dr. Shane Mason to perhaps comment a little bit more on that quickly. I'm just going to give him a few minutes just to sort of highlight, if he can maybe just unmute and explain to us exactly how that pH is maintained. And this is then an example from a biological perspective. Shane, over to you.
Morning.
Morning.
So you want to find out how to maintain the pH on our
Yes,
side?
so one of the questions was just to exactly how it is maintained.
So we use phosphate buffers.
Yes.
The strength of the buffer depends on the sample we're analyzing. So if the sample is very stable, in the case of serum samples, which is very regulated in the body, so there's a very small pH range, then the buffer itself can be quite low in concentration. But the more varied the sample is, the more varied the range in pH, such as urine, because you have a concentrated urine or a diluted urine, then we use a much stronger phosphate buffer to control that. So it depends on what your buffering capacity is required. But yeah, phosphate buffers is what we use.
Okay, so from Dr. Mason's explanation as well as Prof Prinsloo's explanation, you can see that the common factor in the sample prep is then the phosphate buffer that is added. So how your how your buffering system works, I think a lot of you can review your biochemistry textbook, the section on your buffering capacity and how buffers work. So specifically go and look at the functionality or how the phosphate buffer system then works. So the other aspect that was covered yesterday was the fact that I asked Prof whether you could do a dual extraction. And of course, it is possible where you can have your chloroform methanol in different ratios made up as one sort of extraction solvent. And you can, of course, then extract your sample in that way. But Prof emphasized that they do it separately. I can understand why, because if you then extract apolar molecules on one side as well as your polar molecules on the other side, it's a much cleaner extraction because you don't have certain peaks that might be overwhelming or too large. that sort of suppresses some of the metabolites in your sort of apolar extraction and vice versa. So it just makes for a cleaner extraction. Okay, so then the other question that was raised was also regarding, and it was specifically with regards to why a pH of 6 was used. So, of course, this Prof emphasized to you guys that this is a standardized method. It has been developed and fully tested. So, of course, there needed to be some optimization. So, authors would have published this after having tested, the experiment at different pH levels. So, they might have found the same, set of pH 7, pH 7.4, but they probably had better resolution at pH 6. And so there might also be then a reason that, in plants specifically, you might get better resolution at pH 6. That is 100% correct, but that is what I would think happened. So it's not just that they did it at pH 6 and that's how it's done. So obviously prior optimization was done, and so that is why people are now to use it. So in the context of biological samples such as your serums, we usually use a pH of about 7 or 7.4 because that is in the range of what we define as physiological pH. Are there any comments or questions at this point? I just wanted to recap on some of those. Okay. I don't see any comments or questions. And then on the aspects of switching between solvents, again, you would have optimized before you experiment which solvents gives you the best extraction. And so once you have optimized that, the different solvents as well as the ratio of those solvents, you would then stick to that particular method. So Prof placed emphasis on the fact that you should then not switch your solvents once you have optimized, just to sort of assist with maintaining the reproducibility in your experiment. Something else I wanted to highlight for the people that are doing plant work versus people who usually work on, for example, blood-based samples. Usually for serum, we would usually just filter those samples to get rid of the proteins, your macromolecules. And we would then run those samples on the NMR. So of course, your serum or plasma is a sample that's almost sort of ready to run for additional extraction. In the case where you do not have filters, you might, for instance, need to use solvents such as acetonitrile to get rid of the proteins. But in the case of serum and plasma, simple filtering, and then we resuspend that sample in a deuterated buffer and we run that sample. Of course, material like, cells, tissue, plant material, you need to lyse that material in order to get inside of it to get those intracellular metabolites out of that system. So I hope that was kind of obvious and clear. Is there anybody that would like to say something at this point or comment or ask a question? Okay, so what I'll do is I will hand over to Prof Prinsloo shortly.
Good morning.
Prof, I'm just checking. Are you ready?
Good morning.
Morning, yes. Do you have a question?
Thank you very much for organizing this program. But some
Pleasure.
of us were unable to connect yesterday. I wouldn't know if we will not miss too much and we'll be able to follow the class today.
So today we'll be covering some of the basics of NMR and some of its applications. So I think today will be a good recap for you, but we will also post on our YouTube channel the recording from yesterday. So continue to make notes. And if you feel that you sort of got lost somewhere in between still, post your comments on the YouTube channel once you've watched the videos again and we can respond to those.
Okay, thank you.
All right, pleasure. So at this point, I'm going to hand over to Prof Prinsloo.
Okay, good morning, everyone. Yeah, so thanks, Aurelia, for the recap of yesterday. And I think it's important that if there are questions, please post them, keep them. If we are busy with something and you have a question, just note them down, write them down. As I said yesterday as well, it's often that a question is something general. If I miss something, then many of you will have the same question. So please ask them or post them if you're not sure. Let's rather make sure that everyone is on the same page than missing out some of the information. So this morning I am going to do a basics in NMR so that you all know what you look at when you do your NMR, when you get your analysis, to also assist when going to your annotation of your compounds, and to make sure that we don't do inaccurate annotations and that we don't make wrong conclusions. After that, Dr. Shola will do a presentation on the NMR machine itself, as well as other equipment, as well as the integration with other omics technologies or other equipment, just to give an overview of it's not the only equipment that we use, and it's often we assist or we supplement the information with other techniques as well. And then later, the last presentation for today will then be examples. So I'll give examples of a targeted analysis and untargeted, and we'll also give examples on other systems as well, like the soil that we talked about briefly yesterday. You just can get an idea of the type of applications that you can have with NMR, as well as the information that you can get from there. So yes, so let's start then with the basics of NMR. And feel free to post your comments. I will check if I see something, or just unmute your mic if there's a burning question while I'm going through the presentation. Okay. Okay, so there's a lot of writing, but there's a few things I want to highlight here. So this is just a paragraph on how the NMR is functioning. So the principle behind NMR is that many nuclei have spin. So the first important thing is that you have spin. So the atoms are spinning the whole time. And because of that, you can have them electrically charged. So you have a positive and a negative charge. Then if you apply an external magnetic field like you do with the NMR, so your NMR is the strong magnetic field that is applied around your samples. Then you get energy transfer, and you get a base energy, and it goes to a higher energy level, and it can go back to that base energy level. And that loss of energy is then picked up by your equipment. So you apply your magnetic field, you energize, or you transfer energy, and when it goes back to the base energy, that loss of energy that is then picked up, which will then eventually give you the peak on your analysis. And that energy transfer takes place at a wavelength, and that we will see later on where it is on your analysis. And the signal that matches this transfer is many ways, and that then gives you your peaks. So it's important here that you have your spin of your atoms, and when you have a magnetic field, you apply that energy to that atoms. You have a higher energy level. It can drop again back, and you have that loss of energy, and that is then picked up by the equipment. Okay, so what we're going to look at is the spin, how your atoms work, the importance of your equipment with a magnetic field and how it reacts when you apply this magnetic field. Then we'll also look at the chemical shift, which is very important in the NMR, because it's important that you know where on your analysis you find what and what is causing this chemical shift. Then we'll look at splitting patterns, which tells you a lot about what you see and what information you get. And then I'll just briefly touch on 2D. This is a bit more advanced and will be more specific to a specific project and when you get to that level, but I'm just going to show you what the 2D NMR is and the applications of that. So here at the bottom is basically what you saw in the video yesterday. So here's your NMR in a casing. So you put your sample in at the top. It's pulled down into this equipment. Around that you have your magnets, and there's your sample. So your sample is going right to the center, and then depending on the amount of or your hydrogens and the environment around your hydrogens, you get your peaks, which is then sent to your computer, and that's what you get and what you will analyze. Okay, so I talked a bit about, or I talked earlier about the spinning. So we know that nuclei are positively charged, and you have your electrons that's negatively charged, and the spin is determined by the number of protons and neutrons, which is your core. And atoms with odd number of protons will have spin. So you can't do that on all atoms. So we do proton NMR, which is the most easy one and the fastest and the quickest method to use. But you can also do carbon, nitrogen, and fluorine. So those are also applications or different analysis that you can get on your NMR. So, and this is just examples of how it can spin any direction. So you have your center, your nuclei that is positively charged, and electrons that's floating around. Okay, so this is what happens if you apply your magnetic field. You normally have your atoms in all directions, and because you have your because it is positive negatively charged, you have a slight polarity here. When you apply your magnetic field, they're all aligned to this magnetic field. And this is where you're going to apply your energies. So you can get some of them that go into a high energy level and some of them drop. And then there's this field where you apply your magnetic field, where the environment around this proton is going to play a role. So we'll have a look now at shielding and deshielding, but your electrons around your proton is going to either protect this atom from this magnetic field, or if these electrons are pulled away, then it's more exposed, and then you get a different response to this magnetic field. Okay, so what is influencing the type of analysis or the quality of your analysis you will get? So the first thing is your size of the magnetic field. So I showed you yesterday a 600 megahertz equipment. So that is what we use. But you get 500 to 400, 300, 200. You also get bigger ones, 1,000, 1,200. So the size of the magnetic field is increasing your resolution. So the bigger magnetic field you have, the bigger equipment, the more resolution you have, and the more information you can get. Also, you can improve your analysis by increasing the time that it's exposed to the magnetic field. The samples that we run, it's normally 32 or 64 scans, so it takes about 3 or 4 minutes to run one of your samples. If you do not have a strong magnetic field or a strong equipment, you can increase your time to 228 or even longer scans, which then just going to increase your time of analysis. So it's best to find the largest equipment that you can with the biggest magnetic field, because that will shorten your time of analysis and improve your resolution. And the last thing that we're going to look at is your environment of your hydrogen atoms. And this is the basis of your whole analysis and your interpretation later on. So the chemical shift is where you find your peaks on your analysis. And different protons will give rise to slightly different frequencies and measured in ppm. And your analysis will also be measured in ppm. And the effective magnetic field is affected by shielding, or we can also call it deshielding, when the electrons are not around your proton. And this is influencing the magnetic field that is applied to your proton. So for instance, there at the bottom, I've just shown you, for instance, this can be, for instance, a hydrogen proton. And when you have something like an oxygen next to it, which is very electromagnetic, negative, so it pulls the electrons closer, and then you get an exposed proton like this. Normally your electron will be around this proton, but when you have something like your oxygen next to it, it kind of exposes this proton. So the magnetic field is then different on this proton than when you have your electrons around it. So this is what I've been talking about. So we talk about your analysis that you will get like this. Your scale is your ppm. So when you're shielded, so when the electrons are around your protons, they will be around this area here, 0, 1, or 2, and we call that upfield. When you have exposed protons, when the electrons are pulled away, then you get what we call deshield. It's deshielded or downfield. I'm going to give you examples later on of what we mean here, but it's important to just know these words shielded or deshielded, and it's all about the electrons that is either around your proton or that it's pulled away. So if you look at this example here, you have two carbons and your hydrogens on the carbon. So there's nothing that's going to influence the hydrogen atoms here. So this will be a shielded proton. The electrons will be around that. And in this case, you will have your hydrogen or your oxygens next to that. In this case, you will have an effect from this oxygen. It will affect your electrons around your protons, and these will be slightly exposed. And then you have a different ppm reading, and it will then move your peaks on this axis. So just bear in mind the environment around your hydrogens, which is important, which will then have your peaks in the shielded area, or when it's deshielded, it will move downfield. We'll have a look at that a bit later on. Okay, so this is, if you go back to your first year chemistry books, you will see these diagrams. It's a nice guide to use. It's not 100% accurate, and especially when you use extracts and there are other compounds around, and it depends on your type of molecule you're looking at. But this is a guide on where you can expect your hydrogens. So if you have just a long chain single bond carbon hydrogen chains, they're normally around 1 or 2 because it is shielded. There's nothing that affects the hydrogen atoms there. The electrons are just floating around the protons, so they are shielded, and then they are close to the 0. So 0, if you recall yesterday, the T speed that we add to our samples will be at 0. And all your peaks are in reference to this TSP peak. So when you have shielded hydrogens, it will be around 0 and 1 and 2. So when you start to have a different environment around your carbons, it will start to move downfield because they are slightly more exposed. So you have a double bond oxygen, for instance. So it affects the hydrogen, it affects the electrons around your hydrogen. When you have a triple bond, you affect the electrons around it. So now you start to have this deshielding effect around your hydrogen. Same with things like your fluorine, chlorine, bromine, that is pulling your electrons closer to them, so it's slightly exposed. And then you get your different groups, for instance, your aromatics. When you have an aromatic group, aromatic ring, we'll have a look at this later on as well, where your electrons is moving in this ring, and it creates like electron cloud. So then all these hydrogens are really exposed. So then it's moving from 6 to 8 downfield, your aromatics, your aldehydes, carboxylic acids. So this is giving you a good example or a good guide of what you can expect where, and give you also some information already. If you get your sample, if you get your analysis from your sample, this is a nice thing to use to get an idea of what you have in your sample. So this is what I've been talking about. This is your aromatic ring, but let's just have a look at this. So this is, for instance, your proton. This is your electrons around it. If your electrons is there, it is going to shield your hydrogens. So you have a different effect here than what we have here. So here's your aromatic ring. So it's a 6-membered ring. So, and the electrons are moving in this ring, which then creates electron cloud at the top and at the bottom. And you have a concentration of your electrons then in here, which then expose all your hydrogens that's here on the side. And these hydrogens are really exposed because of this cloud that's created here in the center. And that's why you have this really strong deshielding effect on your aromatic rings, on your aromatic compounds. And examples here are your phenolics. So your phenolics all have one of these rings, and your hydrogens are always then showing peaks in the region of 6 or 7 or even 8 because of this deshielding effect. So this is an example of an extract. This is now a plant extract. So you'll see there's your TSP peak at 0. And then you have your ppm, so it's going from 0 to 12. And depending on what type of compounds you have there, your hydrogens on those compounds, you'll have your chemical shifts here. So in the region around 1, you have your single bond carbon hydrogen chains with a lot of shielding. And then as you start to move down, you have your, what we call here, between 3 and 4, your sugar regions, where you have your typical oxygens, or you have your oxygens in your ring. So these are your sugar region peaks. There's, for instance, the water peak, which I talked about as well. So you can suppress that. If it's overlapping some of your peaks, you will see there, you can see it's overlapping some of your peaks. And then we get to the aromatic region. So you can see here, quite a few compounds, which has got protons in this aromatic region. And these peaks are, these hydrogens are really exposed, so they are deshielded. As you can see in this region, you have your aromatic, probably most of your aromatic hydrogen atoms absorbing in this region. The next thing we are looking at is the spin-spin splitting. And this also provides you a lot of information on each of your peaks. So the chemical shift is one thing. This is where it is on your ppm scale from 0 to 10 or to 12. But your spin splitting is looking at each of your hydrogen peaks, and it shows you what the environment of that hydrogen peak was. So that is quite important. So your spin splitting is normally observed between nuclei that are 1, 2, and 3 bonds away. I'm going to show you now. An important here, we talk about chemically equivalent and chemically non-equivalent hydrogens on your compound. So these two things we are going to look at now. So it's depending on how close another hydrogen is to a hydrogen, another one. And then it's important that we look at if it's chemically equivalent or not. Because if it's chemically equivalent, we talked about yesterday about the quantification. So it's chemically equivalent, and there are more than one hydrogen. Your peak will increase because the higher your peak is, the higher your concentration is, or the more hydrogen atoms there are. So this is an example of showing nicely the environment of the hydrogens. So this is your hydrogens attached to a carbon, usually attached to for instance, a chlorine. But these three hydrogens are exposed to the same chemical environment. Remember, this thing is spinning the whole time. So all three of these are exposed to the same chemical environment, and these three hydrogens we call then chemically equivalent, which then mean all three of these will give a peak at the same area or exactly at the same place, and the peak will be 3 times higher because it gives you it's going to pick up all three of these hydrogens. In this example here, you have a chlorine on the one side, and on the other side you have a hydroxyl group. So the environment is completely different on the two sides. So these two hydrogens is chemically equivalent because it's attached to the same carbon, but these two will be non-equivalent to these two because these two are close to the oxygen. So this is a different environment, and that is a different environment. So in this case, you'll have a peak for these two, and you'll have a different peak for these two. And then also they will cause a spin splitting pattern. So this hydrogen is affected by these two, and it will then show a different splitting pattern. The same with this one. This one is going to see these two hydrogens, and they will have a different spin splitting pattern for this one. So just bear in mind now the two things that we talked about. The one is the chemical shift, which talks about the environment around your hydrogen on where it is on this ppm scale. And then we have a look at the hydrogens that is next to it that's going to cause a splitting pattern. As you can see there, it's 1, 2, 3 bonds, 1, 2, 3 bonds. So these are close enough to have a splitting pattern or to have an effect on the peak of for instance, that's hydrogen. So this is what your peaks will look like once if you have a splitting pattern. So if you have just one hydrogen, there are no hydrogens around, you just get a singlet. When you have a hydrogen close by, which is non-chemically equivalent, when it's 1, 2, 3 bonds, so this hydrogen will be seeing this one that's close by, and it will split it into a doublet. We call it a doublet. The two peaks will be the same height. And we'll talk about this J constant later on. This is very specific for a specific compound. Then you have, if you have, for instance, 2 hydrogens, it's 1, 2, 3 bonds, 1, 2, 3 bonds. So these are non-chemically equivalent to this one. So when this hydrogen is seeing these two compounds, it's going to split into a triplet. So you have 2 shorter ones, and the 2 in the center is on top of each other, so it's double the height. So you have this typical shape here, this triplet. And when you have 3 hydrogens around a hydrogen, all of them are 3 bonds. So this hydrogen is seeing 3 of your hydrogens next to it, and they call it quartet. Okay, so they form 4 peaks. But this pattern is only for this 1 hydrogen. So it's not 4 hydrogen peaks, it's 1 hydrogen. And that's why we'll later on look at these J constants as well, because this will be the same if it is if it's belonging to one hydrogen. The same here. These 3 peaks is for this 1 hydrogen. It's not 3 hydrogen peaks. It's only 1 hydrogen peak, but it's split into a triplet because it's seeing the 2 hydrogens on the side. The same in this case. This is the pattern for 1 hydrogen. And it's because it's seeing that 1 hydrogen splitting it into a doublet. Okay,
Prof,
yes.
Can I just add something? Just take note on the previous slide that Prof showed you guys. Hydrogen is linked to a carbon. So the hydrogens that are visible are usually linked to your carbons.
Maybe at this stage, are there any questions? Well, we have a bit of a pause here.
Prof, I have a question, but I'll bring it up later on because it has to do with the databases.
Okay.
So it's a question from yesterday.
Okay, I'll also make a note here about that. Okay, so let's have a look at some examples, and we can come back to this later on if you don't understand, if you missed something. But this is very important that we have the 2 basic concepts here of chemical shift, which we talk about the movement of your peaks on this scale, on this ppm. So you have a deshielded one and a more deshielded one. So the chemical shift talks about where on this axis your peaks are. And then we talk about the splitting pattern, which then talks about the pattern of this hydrogen. So this is 1 hydrogen, and this one is split into a doublet. And this is 1 hydrogen that is splitting into a quartet. So this is what the structure looks like. So you have 3 hydrogens, and as I mentioned earlier, your NMR is quantitative. So this peak of these hydrogens will be representative of 3 hydrogens. So this will be much higher than when you just have 1 hydrogen. And in this case, these 3 hydrogens are chemically equivalent. So they will not form a splitting pattern amongst each other, but they see this hydrogen here. So each of these will be split in a doublet, and there are 3, so your peak will be 3 times higher than when you have just 1 hydrogen. But because they are chemically equivalent, they will all absorb at the same place or show this peak at the same place, and all of them will show the same doublet. So this hydrogens here then shows a doublet, which is quite strong because of the concentration or the amount of hydrogens that is seen here. And this we talk about the integration or the integral, the height of your peaks and the number of protons that is represented by this peak. If we look at this example now, this is this hydrogen. So you can see it's next to a chlorine, which is pulling the electrons closer. So this hydrogen is much more exposed or deshielded, and that's why it's moving downfield. So at around 6, you find this hydrogen peak. So this hydrogen peak is seeing this hydrogens next to him, and there are 3 of them, and that's why it's forming a quartet. So once again, these 4 peaks that you see here, it's only representing that 1 hydrogen. So it's not 4 different peaks here, it's actually 1 peak that is in a splitting pattern, and that pattern is then representative of the 3 hydrogens that it sees next to it. Okay, so I'm just going to emphasize again, we have 2 things here. The one is the chemical shift, so where it is on your ppm scale, and that is as a result of what is around your hydrogens. So these hydrogens are linked to a carbon, linked to a carbon. In this case, this hydrogen is attached to a carbon, linked to a chlorine. So this environment here is much different, and that is why you have this chemical shift of these 2 peaks. So that's the first thing, is the chemical shift. And secondly, we talk about the splitting pattern. And the splitting pattern is the pattern that you see, and that is as a result of the hydrogens around it. So just be clear about the 2 differences, different concepts here, the chemical shift and the splitting pattern. This is another example showing clearly the difference in your chemical shift and your splitting patterns. So let's have a look at this, this one here. So those 3 hydrogens here, it is attached to a carbon, attached to a carbon. So these hydrogens here are not really exposed. They are, all the electrons are around them, so they are shielded. So we expect this peak then to be around 1 or close to 0, because it's not exposed, it's shielded. This carbon, these hydrogens is attached to a carbon, which is next to a oxygen. So in this case, again, this oxygen is going to pull some of the electrons closer, and these hydrogens are then more exposed, so they are deshielded, and this peak is then moving downfield, and we expect it around 4, between 3 and 4, around this area, because of this oxygen next to it, and it's exposing these 2 hydrogens. In this case, your hydrogens are attached to a carbon, attached to another carbon, but on this carbon you have an oxygen. So once again, also it's the oxygen there is going to influence your electrons, but not as much as this oxygen that's attached to your carbon directly. So in this case, yes, it is going to affect it, but it's not going to have such a big effect on this. So it's moving slightly downfield to around 2, but it's not moving as far as this one, which is directly next to your oxygen. Okay, so this gives you a bit of an idea about how the chemical shift is working, and where you will expect your peaks when you are looking at your structure. Then the second thing is, then we can look at the splitting patterns. So these 3 hydrogens here is next to a carbon with 2 hydrogens. So if you have 2 hydrogens, you're going to have a splitting pattern of a triplet. So once again, these 3 peaks is only representing 1 hydrogen, and the height of this will represent the number of hydrogens that is shown by this peak. Okay, so it's a triplet, and it's a triplet because of the 2 hydrogens next to it. In this case, you have 3 hydrogens, but there are no hydrogens next to it. These 3 hydrogens are chemically equivalent, so they are not going to split each other, and they will form a peak or show a peak at exactly the same place. So the magnetic field that's applied here will be equal to all 3 of these hydrogens. And there's no hydrogens close by. The closest, or the bonds 1, 2, 3, 4, 5. So that hydrogen is too far from this one to have an effect on this and to create a splitting pattern. So these hydrogens will just form a singlet. And because there are 3, you'll have a strong peak there because it's representative of your 3 hydrogens. So once again, indicating that your quantitative effect here, that there are 3 hydrogens and not only 1. So you have the strong singlet here around 2 because of the oxygen next to it. So it's a little bit deshielded, not a lot, but a little bit. So it's moving a little bit downfield, but your 3 hydrogens is forming 1 singlet. and a strong singlet because the 3 hydrogens are exactly at the same place, forming a peak at the same place. Then the last one we are going to look at is this one. So next to these two, we have 1, 2, 3 bonds, 1, 2, 3. So these 3 are close enough to have a splitting pattern on these hydrogens. These 2 hydrogens are chemically equivalent, so they will be having a peak at the same place, because this whole thing is spinning around. But each of these hydrogens will see 3 hydrogens next to it. So this hydrogen will form a quartet as well as this one. So this peak here is representative of 2 hydrogens, but because of the splitting pattern, it is slightly smaller. But the splitting pattern is a quartet because of these 3 hydrogens here. Okay, so once again, just see the difference here between the chemical shift. The carbons here, that is not next to a oxygen or something that is going to expose the protons. So they are shielded and they are then upfield. This one is right next to an oxygen, affecting the electrons around this proton. So it's moving downfield, it's deshielded. And these ones is something in between. It's attached to a carbon, attached then oxygen to that carbon. So it's slightly affecting the electron fields around here. So they move slightly downfield, but not as much as this one that is next to an oxygen. Okay, so I'm not going to spend a lot of time on the coupling constant, but we talked about that a bit earlier. So, so the coupling constant is J, and that's a measure of the interaction between a pair of protons, and that's the distance between these peaks that you have in a triplet or a quartet. And that is specific then that you use to make sure that it's not peaks of different hydrogens, but it's all belonging to one, and it's belonging to a splitting pattern. So this is just showing you again. So so there's your J. So you measure the difference there, the ppm, and that is specific for a specific splitting pattern. The last thing I just want to mention, I'm not going to go into detail, but you might maybe see this in papers and you will wonder about this. So the ones that I've explained to you now is one-dimensional NMR, but you can also get two-dimensional NMR, where you have your one-dimensional NMR on this side and on this side, and then it looks at the interaction of your hydrogens. So there are quite a few. I'm not going to go into detail, but when you get to this level, you'll know there are two-dimensional NMR, and it can also assist you in when you're not sure about a compound, when you look at annotation of your compounds, then you can use your two-dimensional NMR to get a better idea of your compounds and to confirm. You can also confirm your identity of your compounds with this. Okay, so in general, we talked about this yesterday as well. NMR is a very versatile technique. It can be used on any samples. We talked about the bacteria, the soils, your plant extracts. You can use serum, you can use urine. So any sample you can use in an NMR, because it's not injected into the system and going through the system. It's staying in the NMR tube. It's non-destructive. So you can put any sample in there, and the magnetic field is applied around your sample. So it's not, so your sample is not going out of that tube into the equipment. So you can use it on any sample. It's unfortunately not as sensitive. We talked about that yesterday. So your chromatographic techniques like your LC and GC is much more sensitive. And that's why we often use NMR with other chromatographic techniques as well. So once you have your answers or you have your compounds annotated, you will then confirm with some of the others, with some of the other techniques, and also it will also supplement your information. when we start to look at, for instance, a metabolic pathway, you have your compounds annotated with NMR of identified, and you identify a specific metabolic pathway, then often we find that your GC or your LC will give you more information of the compounds in that specific pathway. But then you've already identified your pathway with your NMR, and then you can supplement your information with other techniques. So the magnet size is important. The bigger magnet you have, the better resolution and the better your samples will look like. If you do not have a big one, you can improve your resolution by just running it longer. But as I mentioned, that will increase your time, analysis time. So bigger equipment is just a bit easier to use. You can, as I mentioned, increase the number of scans. So with the 600 megahertz, we normally use 32 or 64 scans, which is not a lot. So that's why it's quite fast to run your samples, about 3 or 4 minutes. So if you use only 32 scans, for instance, you can run about 20 samples, 15, 20 samples in an hour. So within a day, you can run easily 100 or 150 samples. And then your NMR is quantitative, so that's a very nice feature of the NMR. So the height of your peak is an indication of the concentration of that hydrogen in your sample. And you don't need to rerun your sample. Once you have your sample analysis, even if you don't look at the concentration differences initially, you can go later on and can work on that, on the concentrations, or find your concentrations of a specific compound or the peaks by using the same sample. So yeah, it's just an example of how your resolution can be improved. So you'll just see splitting patterns better if you use more scans or if you use a better equipment. Okay, so in conclusion, we started with looking at the spinning of your atoms, and it's because of that spinning that you can get NMR analysis. And because of that spinning, you can apply the magnetic field, and you have different responses of hydrogens in a different environment. So your magnetic field is applied, and that can cause an increase in energy levels. And when that energy level drops back to its original level, then that loss of energy is picked up by your equipment. And obviously, this energy level differences will be affected by the magnetic field that is applied. And if it's shielded or deshielded, this energy levels will be affected. And that's why you get a difference of your peaks on your ppm scale. So if it's shielded, the effect will be less than when it's deshielded. The magnetic field will have a bigger effect on your protons. So this is just for example here. If you have something like an oxygen, which is pulling your electrons close, you'll have an exposed proton. So this deshielded one will have a different magnetic field exposed to this proton than a proton where the electrons are around. So the frequency is captured, which then eventually give you your peak. We talked a lot about the chemical shift. So, and please, again, I'm going to emphasize it again. Please see the difference between the two. The chemical shift is where this peak is on this ppm scale, and that is because of the shielding or the deshielding effect. So a peak that is at 0, 1, or 2 is where there's no deshielding effect. It's shielded. And the more there are things around that can affect the electrons around your proton, it's moving downfield because of the deshielding. The electrons is away, and that magnetic field is having a different effect on that proton because the electrons are not around. Then the second thing is the splitting pattern, and this tells you something about the environment of your hydrogen. So it's not going to affect the place on your ppm scale. It's not going to affect the chemical shift, but it tells you something about that hydrogen, if it is around other hydrogens and how close they are, and if there are the amount of hydrogens around that. So it tells you something about the environment of that hydrogen in terms of other hydrogens that's close by. Then the coupling constant will help you to make sure that the splitting pattern belongs to a single hydrogen and that it is not peaks of different hydrogens. And the height of your peaks is an indication of your concentration. We also call it the integral. Is an indication of the number of hydrogens in that peak. So the higher the peak is representative of more hydrogens. And because of the equipment is not as sensitive, when you have a splitting pattern, for instance, this one, your peaks get very low. So then you can lose some of your information. So then you often have to supply or get other analysis to help you with making proper interpretations of your data. But that also tells you something about your compounds in your sample. Just something on the non-sensitivity of your NMR. Yes, it is a disadvantage, but for me, it's often also at an advantage because the equipment is not that sensitive. So if you find differences in your NMR profiles, it means that that is definitely a strong difference between your samples. You won't see a difference on your NMR if it is not a big difference between your different samples or your different treatments. So the moment you see something on your NMR, you know it's a very strong or a very pronounced difference between your samples. And you should then definitely look into that further. You might then also lose some of your information because the sensitivity is not that good of differences that are there between your samples. But the ones that you see on the NMR, you must definitely investigate further because that shows that there's definitely a big difference between those peaks that you find between your different samples. Okay, so that is the end of this presentation.
Okay, thank you for that, Prof. I see there are some questions in the chat. I think you just responded sort of to Muhammad's question, because Muhammad was asking about the fact that NMR, if it's not that sensitive, why don't we just carry on using GC or LC? Yeah. And I think that the fact that not all, remember with metabolomics, you are not going to sort of detect your metabolites using one particular method. You can never sort of fully detect everything. What you are doing is trying to get a comprehensive metabolic profile. You will get everything from there. So the best, remember, even if you do use GC or LC, there's always a compromise. GC, for example, you prepare your sample in such a way that it's volatile. So in that case, you're using specific solvents, and in that case, you are then losing certain metabolites on the choice of your solvent. So you have to look at what is your research question and which method will then work best for what it is that you're trying to answer. So while NMR is not as sensitive, it is reproducible.
Yeah.
So I don't know if you just want to add on that.
Yeah, so another thing for me for the NMR is, for me, the NMR is a bit of it's a simpler technique, and the interpretation is a bit, it's more definite. You have less chances of making inaccurate or incorrect conclusions from that. Especially if you are not well trained in analytical techniques. The LC-MS is a very complex and difficult equipment to understand, and if you do not have a lot of experience on LC-MS, you can easily make wrong assumptions and wrong conclusions from the data there, especially in annotation or identification of the compounds there. So, so if you're not very experienced, the NMR, if you do not have a strong chemistry background, if you haven't done first year, second year, third year chemistry, but you want to use metabolomics, then NMR is for me a very good method to use, because it's easier to interpret. You do not have this masses of information that you have to process. As I say, that the peaks that you have there that are different are really strong differences between your samples, and there are a few checks that you can do on your NMR that we'll have a look at tomorrow to make sure that you do not annotate incorrectly. And even if you are not 100% sure, you'll get a good idea about, for instance, the class of compounds or the structure. You can get a good idea about the structure, maybe not 100%, you'll have to maybe use LC-MS, but you already have a good idea about the compound that you are looking for. And there are ways to check that you are not making incorrect assumptions or conclusions. But when you get to the compound level, the NMR is for me an easier method, especially for students that do not have a strong background in chemistry. I often see a lot of mistakes on LC-MS and analysis where people annotate or identify compounds, which is based on previous information and which is not, you can make a lot of differences there if you do not know what you do with the LC-MS, because it's a very complex analysis that you get. You have a wealth of information. Because it's so sensitive, you get thousands of compounds that you have to look at. So, so with the NMR, it is for me a simpler technique to use and to start with.
Yes.
And once you get an idea of what you have, then you can supplement with your LC-MS and data or your GC-MS. And when you combine these together, then you get a better idea of what you have. But the basic work you've done is on a simpler technique. So that's a benefit for me also with the NMR.
Okay. I'm not sure if anybody else wanted to comment. Dr. Shane, did you have additional input? Okay, I don't think so. I see one of the hands that are raised. Anton, please go ahead and unmute.
Hi, Prof.
Hi.
Morning.
So I know with LC-MS it's difficult to kind of distinguish between isomers. Well, if you have a standard, you can, but if you don't, you can't. So can that be distinguished between in NMR?
Yes. Yeah. You see, the environment of your hydrogen will be completely different in your isomer. So there you can pick it up. Yeah. See, so that's what we talk about. Often you have to use a combination of the techniques to get proper answers. And many people forget about the NMR, but it's a very useful technique to supplement information and to get additional information on for instance, your LC data.
Okay. Is your question answered, Anton?
Yes, thank you very much.
Okay. Dr. Lufuno, yes, you can unmute.
Yes,
Oh,
I wanted to just say that. Please. I wanted to just say that they can use NMR as a first stop. You know, if nothing works, then you can always go to the next one. Because also, if it's for example, if it's a PhD study, you wouldn't want the external examiner to be asking questions or okay, so you use LC-MS. Where is the result? Where are the results for NMR? You know, so you rather just have everything NMR, LC-MS. If you've got like what Prof Oril said, I'm sorry, I'm pronouncing your name wrongly, but what Prof William said, that you know, different analysis tools are used, or softwares are used for different compounds. For an example, you cannot use for GCMS, so you're going to have to be using it for volatile compounds. You know, so it has to do with what are you looking for in your or what are your objectives and what are you looking for. So maybe use NMR as a first bus stop, and then you can always fly or stretch your wings to LCMS and GCMS.
Also,
you,
yeah, I also forgot to mention also, for instance, sugars is not easily detected by any of your other samples. So NMR is very good, for instance, for sugars. You can get a lot of information on your sugars, your combination, your composition of your sugars in a sample, which is not easily detected by some of the others. So there are certain groups of compounds that you can get very good information from your NMR. And then once again, what you are looking for, and it depends on your biological question. And by supplementing, you get a better idea.
Yeah. So in our case, Prof, I can comment, for example, the serum samples, like I said, unlike the plant material, we don't necessarily have to light anything. We do filter the serum and we just add a buffer and we run that on NMR. So in that case, we're not really doing anything much to the sample. We're just trying to detect as much as possible to what is in that sample. So that sort of gives us a comprehensive profile of what is in there. And then we can go further. If we see a whole lot of, amino acids that change, then we can go targeted and use something like ALSI to now better profile those amino acids. But is a good starting point, especially if you don't know what specifically it is that you are looking for. And just like Prof said, GC, for example, doesn't resolve your sugars as well. So then it's better to then opt with NMR. So you have to also understand the nature of your samples and the objectives of your experiment.
Also, yeah, as you mentioned now, for instance, the NMR, if you do not know the specific amino acid, you get an idea about the group of compounds. The same with the aromatics, for instance, the phenolics, your flavonoids. They're all absorbed in that area of 6 and 7. So if you have a huge difference between your samples in that region, then you already know you can target your phenolic compounds. And then if you go to the LCMS, you go specifically and have a look at your phenolics. So the NMR gives you a good idea, even if you don't know the specific compound name at that stage. It gives you a good idea about the group of compounds that you are looking for, which is very, very helpful.
Okay, so I think that basically covers Mohammed's question. He had another question, that is, what is the cost of one sample compared to, for instance, running LC or GC? I don't know if you can provide an answer with respect to UNISA. Shane can perhaps just reply to our chat and give an indication of pricing at Northwest University.
Yeah. So we don't really charge per sample. So what we do is you buy your own equipment, your own solvents and so on. So I can just give an idea about the cost of the solvents. So the solvents is not that expensive, except methanol. Deuterated methanol, extremely expensive. So for the chloroform, I think it's about 1,000 Rand for 100 mils. And from 100 mils, you can run about 70 samples. So the cost is not that expensive. If you then use methanol, it's quite expensive. 100 mils of methanol cost about 7,000. Your water is also not expensive, about 1,000, 1,500. So you can run about 120 samples on a 100 milliliter bottle of methanol, because you only use 750 microliters and then 75 to 750 microliters of water. So then your samples are a bit more expensive, but because you don't need other extraction solvents, that is the only solvents you need. So it's a direct extraction, so you use that immediately, so you extract directly from your plant material. So for instance, if you use chloroform and you buy a bottle of 1,000 rands, you run 120, about 80 samples from there. So, yeah, so that gives you an idea about the cost of what it will cost you to run your sample.
Okay. So Mohammed, just check the chat. Dr. Mason will, for instance, give you an indication of the pricing when you run samples at Northwest University. But keep in mind that, you know, it's not set in stone. There are agreements that can be reached, for example, where a student might be shared if you, for instance, collaborate with, you know, one researchers, where, you know, that pricing can be adjusted and you guys discuss how the cost of the project will be handled. So it's not necessarily set in stone, but there are guidelines as to the pricing. Okay, so I'm just going to move on. So there's a request. Could you also do a GC workshop, please, so that we can get good ideas on different methods? Also, Muhammad, if you subscribe to our YouTube channel, you will see that we've already covered a workshop, I think, on LCMS. So this one is on NMR. We will try and work in some of our future workshops. We will have a symposium later this year in October. So we will send you guys a save the date or reminder just to keep those dates open. But that will just be a symposium. So do keep that open. There might be some GC work that's highlighted during that symposium. He's just saying that he's happy with some of the responses that he's received, Prof. Eugene, I'm just trying to move through the chat. Okay, there was a question about PPI to that, saying it's parts per million. Dr. Shane has also replied there saying each platform, whether GCMS, LCMS, or NMR, can identify, quantify unique metabolites. There is no one instrument that can see everything. NMR is the most repeatable and reproducible platform compared to GC and so that just reiterates what I said earlier. If I move on, I hope I pronounced the name correctly, but I'll go with Bultosa. The question relates to proton or C13 or proton NMR. Which one is more useful in terms of identifying metabolites?
Yeah, so I think you can use any of them, although your proton is your most common one. Your proton and your carbon is just taking much longer. So we do not really use carbon NMR for metabolomics because you use normally large sample numbers. So then it's better to use proton. It also gives you a lot of information, I think, in comparison to your carbons. But we often use carbon then in addition to your hydrogen if you get information and you want to have more information on your sample. So your phosphorus and your nitrogen ones of fluorine are not really used often because you don't have that often in your samples. Most of the compounds we look at are mostly carbon and hydrogen, but you can use that as well, but that is not really commonly used.
Okay, I think the phosphorus NMR, Prof, if I'm not mistaken, is used a lot related to energetics. Right?
Yeah.
Okay, so I think the conclusion there is that you mainly then use your proton or your carbon NMR because of the abundance of those.
And also you don't know what you're looking for. So once again, so your proton NMR is giving you a profile. You extract as much as possible as you can, gives you a profile, and you're going to compare your profiles. And there you want to have as much as possible information to be compared to each other to see if there are differences or not. So that's why we use the proton. It gives you a good profile of your sample.
Okay. I see Dr. Mason has added saying that proton NMR is well suited for metabolites of high abundance, primary metabolism, and for untargeted analysis. So no to little change in your original sample. And this provides robust metabolomics data with a high quality. Okay, I see again, Muhammad, is there any book that you can recommend to help get a good understanding of the process of NMR?
I go back always to my first year chemistry, organic chemistry textbook that I used in first year. It's easily explained there. It's just a few pages. McMurray, that's the one that we used. McMurray Organic Chemistry. So I always go back. Some of these pictures that I've used is coming from there because it's well explained. It's easy to understand, and then if you want to go a bit more in detail, then go and Google. But I would say for the basics of NMR, go and use a first year organic chemistry textbook. It's only a few pages, but it gives you quite good understanding, or it's well explained, and the examples there are very nice. It's examples that you can understand, and to bring the concepts of, for instance, chemical shift and splitting patterns and so on. So so the first year chemistry books is a good place to start.
Okay. In addition to that, Muhammad, I have a book that I received from one of my supervisors. I'll post the detail. It says Modern NMR Spectroscopy. Obviously, this was published a good while ago, but it's one of the standard textbooks that the prof actually used, where I trained a little bit on NMR. Details then of this book on the chat as well. Okay, so I'm just checking to which other question. To which molecules is NMR limited to? Can it do proteomics?
Yeah, so your proteins will not be extracted by the method that we use. So this is just for your molecules. So for instance, your amino acids, yes, you can actually identify many of your amino acids by NMR because they have quite distinct peaks. So many of your amino acids can be identified with your NMR, yeah. But not the proteomics is a completely different process.
Yeah. So, Francis, just to add to that, to Prof's answer, remember with NMR, when we run biological samples, what we do is we actually filter the samples and then resuspend them in a buffer. So what that filtering does is that the filter removes macromolecules or your proteins. So remember, metabolomics, metabolites are small molecules. So we cannot necessarily do proteomics because we've removed proteins. And also when you extract your sample using solvents such as your acetonitrile and methanol, those actually gets rid of your proteins as well. So metabolomics is small molecules. Okay. Yes, so definitely amino acids, but not the actual proteins. There are instances where you have NMR analysis done on lipoproteins, for example, but then that's a completely different sequence that you run on your system. I don't know if you want to comment on that, Prof.
No, I'm not familiar with that really.
Okay. Dr. Mason has replied again to one of the questions. He said, please go to Google Scholar and search for NMR Metabolomics Review. There are plenty of papers that describe the topic some more. Okay.
Yeah, once you have the basics that we will run through yesterday, today, and tomorrow, then if you go to a paper, you'll have a better understanding of what they talk about, and then you can start to supplement the information. But once you're done tomorrow, you'll have quite a good grasp of NMR metabolomics, what it entails, what you do, how you do it, how to interpret. And then if you check those review papers, you will have a good grasp of what they talk about there.
Yeah. Dr. Mason has also reiterated that metabolomics protocols remove proteins. You cannot analyze proteins, but proteomics labs use NMR setup to specifically analyze proteins. I guess that was amino acids. So the next question, Dr. Mason has also added, I'm just sliding through these comments. NMR can detect and quantify amino acids, but if amino acids are your target, then metabolomics is better. So then you would go to LC or GC. Proton NMR is well suited for metabolites. Oh, I think I read that one already.
Yeah, maybe the question there on untargeted. Yes, I will show you later this afternoon. I will show you examples of an untargeted as well as a targeted example. So yes, you can use it for untargeted. As we mentioned earlier, you can get a good information on the type of compounds in any case that you have in your sample. So it's actually also a good method to do untargeted. Yeah.
Is that your response to the COSI one?
Yeah.
Okay. There's also, coupled to that, are there any limitations compared to your proton NMR?
Yeah, so we discussed that your sensitivity. If you have overlap of your peaks, you get that often. So there are ways to then look at that later on to do a fractionation or to make then different extracts and run them to partially purify your extract or so. So there are limitations. We mentioned all techniques have a limitation. So you have to decide which one work best.
Okay, then we have a link that is posted in the chat. The Center for Human Metabolomics, this is at NWU, has the price for obviously the services that they offer. Keep in mind that the prices with CHM are for full service only, meaning there's no collaboration. But if there's a collaboration, obviously we discussed that as I've mentioned before. The YouTube link will be posted by Anza Oke. Just check the chat for that. Mamoho, perhaps Prof Prinsloo can better reply. Okay, Dr. Mason just replied to that. I think you responded to that question already. Rochelle Bester also mentioned a nice book, Principles of Instrument Analysis. She's also listed the authors. So for those of you who are interested in reading a little bit more about NMR, Principles of Instrumental Analysis. Dr. Mason has also posted an ebook on NMR metabolomics. And then there's just a few thank you notes. I think we have covered most of the questions. Have I missed anything? You have to just raise your hand and then ask. Nothing? Okay, so let me just check.
Sorry, please, before you go.
Yes.
Sorry, please, how can I preserve samples that I want to send down for metabolomic analysis? Maybe
Can
possibly.
Can you just
Hello?
repeat that again, Kazim?
Yes, how can I preserve samples that I want to send down to maybe NW for metabolomic analysis? I'm here in Nigeria.
What sample types are those?
Oh, either serum or tissue. Serum and tissue.
Okay, so usually you would have flash frozen your tissue, or
Yes.
you would have stored your serum at -80. So what you would do is you would arrange with a courier company, such as the ones we often use is World Courier or BioCare, and your samples are usually transported on ice.
So please, I would like to have details of such the companies so I can link up with them.
You mean
Or I
the
can call you-
companies that do the transport of the samples?
Yes.
Please post your email address in the chat, and then I will send that to you.
Thank you, Claudia.
Pleasure. Was there any more questions?
I see there's one about the database now.
Oh, the database question. Go back to it because it was posted in yesterday's session.
Yeah. So databases. If you do human metabolomics, it's easy because there's nice databases for that. For plant metabolomics, not as much. You have to search a bit. What I mentioned yesterday also, we commonly use the human metabolome database as a good start because it provides, for most of this, for many compounds, it provides the NMR profile. So if you are not sure about a compound or you have an idea about something about a compound, then you can go to the human metabolome database and check the NMR profile, and then you can match it with the peaks on your NMR. So that is a good place to start. If it doesn't exactly match, you get an idea of what you have and what it might be, and then you can start search further in publications for NMR profiles. But we use that often. Kenomics we also use, but that's also based on the human metabolome database. So there are a few databases. Maybe for the human ones, you have better ones to use.
So I just wanted to add to specifically for Kenomics. How I understand it was developed is that obviously they have known standards that they've run that's in a library, and so you just run yours against those. Most often in your, and then also by the actual pure compound or synthetic compounds, you run those according, under your conditions, save them, and then every time you have samples that you need to run, you can match them against those. But we also often use the NIST library, Human Metabolomics Database, as well as the NIST library to sort of peaks against. So the question from yesterday, Prof, was specifically with, do you use data from biological magnetic resonance bank, from the biological magnetic resonance bank?
No, we have never used that.
Okay. So yeah, I've also not used that before, so I'll look into that. But yeah, that was your question that was posted by Neves Flores. Okay, so I do see that there are some more questions that have been posted in the chat, Prof. Which technique would you prescribe for protein analysis? I think Dr. Mason said, or he replied saying LCMS.
Yeah. Yes, but then you will do proteomics. So then you go to the proteomics side.
Okay, then what is left here? One posted. Dr. Mason has also posted the human metabolome database link. Mohammed again has a question. I am working on the effect of medicinal plants on cancer cells. Mohammed, you haven't completed your question. I'm working on the effect of medicinal plants on cancer cells. He used GCMS.
Yeah.
Okay.
You see, the GCMS will target your volatiles. So So yeah, so if the idea was then to work on volatiles, then yes, but if it is not really focused on volatiles, I think you're going to miss a lot. There might be many compounds that you miss out when you look at only GCMS. So, but yeah, it's difficult now to understand what you're doing there. So, if you maybe put a bit of information, otherwise I can also provide my email address. Then you can email me. Then we can have a look at polar compounds. Yeah, then you'll have to use other. For instance, then your polar extraction for NMR will give you a good idea about the polar fraction of your extracts, but also then LCMS. But your GC will not be a complete profile. You will be very selective.
Definitely. Any more questions? I think we've covered a lot for the session. So what we will do is the next session starts at 11. So just to recap then, in the first session, we basically covered just the principles that you need in order to understand NMR metabolomics, as well as just the basics to help you interpret your data. So what Prof looked at specifically was chemical shift, as well as the splitting pattern then of your hydrogen. So there you would have heard words or concepts such as shielding or exposed, upfield, downfield. So make sure when you are reading the literature or learning about NMR, especially the people who requested the books, that you familiarize yourself and then just make sure that you understand those basic concepts. So that was some of the key concepts that was sort of highlighted in our first session. So the next session that we will cover basically deals with more of the applications of NMR, and what will be highlighted there is how NMR can also be coupled to different technologies, to other technologies as well. So I'm going to hand over to Dr. Budere.
Okay, thank you. I believe you can hear me.
Yes.
Yes, I can.
Okay, let me just try to share my slide. I think I'm sharing for the first time on Teams. I've been using other platforms though. Okay. Is it trying to open?
Yes, something is happening.
Yes, it's showing.
Okay, I'm not yet winning. Do you think I need to first make my PowerPoint to be in presentation, PowerPoint presentation, and then share? Because I can see that only the first slide is showing at the moment.
Yes, I think you can do that.
Okay, just a moment, please.
So while Dr. Budere does that, just a reminder, guys, I've posted the Metabolomics YouTube link. Please subscribe to it. So when we post the videos from the past 3 days, you will then get a notification. So once you click subscribe, you will then get an automatic notification once we upload the videos.
I can maybe just also give a bit of background. Dr. Budere is a postdoctoral fellow at UNISA. He's a natural product chemist. So he joined us to specifically help with annotation, identification of compounds, and also did quite a bit of metabolomics work. So if there's any of you that needs to have assistance with a natural product chemist, can always contact Dr. Budede. That is actually his field of speciality.
Okay, I think I just need a bit of help to have this slide.
Yes, Dr. Budede. So to the left, I think of your on your screen, there will be a little note that says end presentation.
Okay, stop presentation.
So you can stop presenting, and then you can just go onto your desktop and maybe convert to presenter mode, and then you share again.
Okay, yeah, I've put the slide in presentation mode now. So I need to just go to share content. Will that be correct?
Yeah, open your share tray. That's what my label says. Yeah, you can share content.
Okay. I have presenter mode, content only, and then screen. No, sorry, no. Okay. Window screen. Okay.
Yeah.
Okay, I'm sharing my screen now.
Yes, we can view. I can see application of NMR in metabolomics.
Oh, okay, good. Sorry for that. Okay, thank you very much. I've been introduced already. Olushola is my name, and within the time frame, we see how to do some justice. I will try to consider the audience as either people with some background in NMR, and also I will still put into consideration those that really haven't got much background in NMR. Thank God for Prof Prinsloo. He has given a lot of background. There will be maybe a few overlaps, but I won't be stressing too much on areas that Prof has covered. Let's see to what extent we can go with the application. Okay, so as you can see on my screen, basic principles behind NMR have been discussed. I still want to stress the fact that the nucleus is very important. We are dealing with the nuclei of elements of atoms that are in our organic compound. The magnet is very key, because without the magnet, the spectroscopy is just empty. So the bigger the magnet, the more you have 800 megahertz, 600, and that gives you better resolution, as you have already had it. So with a bit of touch on spin, you have also heard about spin. There must be ability of that particular nucleus to spin, especially the proton part of the nucleus. Okay, so there must be spinning. Then you have your spectrum. And then we talk a little bit about the chemical shift, which Prof has also emphasized. So this method depends on the magnetic property. We've already mentioned that. I will still be giving you additional nuclei that can be used apart from the proton, the hydrogen one, 13 carbon and 31 phosphorus. Okay, it is the most frequently applied in metabolomics. The reason being that it's easy to acquire the data. You need less time with appreciable amount of quantity of your material. You can easily get your 1H NMR spectrum. And for metabolomics, we'll be dealing with a robust data. You can have up to 100. Sometimes it can even run into thousands, depending on how big the experiment is designed. So please take note of that. For carbon, if you are running a proton for like 5 minutes, sometimes 10 minutes, you might need an hour, if not more, 2 hours for carbon 13. So that is about that. So the technique operates by placing a liquid, so I won't be stressing on that. Prof has given us that details. Okay, the sample is paused with a range of radio frequency covering all possible energies required for exciting the selected type of nucleus. So there must be excitation. So you see, when you put an atom in between magnetic field, you are creating that resonance. So there must be excitation, which we already have some backgrounds into. So let me just give you these details. 1H, 13C, 15 nitrogen, 19 fluorine, 31 phosphorus are common nuclei that are being used in NMR. They are very, very applicable to these ones. And you will notice that they do have odd mass number or atomic number. And that makes them really the nucleus to be, the nuclei to be available for this type of spin. Okay, what happened is that the nuclei absorb energy at different frequencies depending on their chemical environment. And then release that particular energy, which is measured. So then we have the formation of free induction decay, the FID. People that have been dealing with this NMR data, you will notice that you always bring in your data, the FID part of the file that you obtain, that you now put into your software for pre-processing and the like. So the FID is converted. It's converted from a time domain dataset to a frequency domain using Fourier transformation. And you have your spectrum, which is constructed as chemical shift. A chemical shift, effectively, the absorption energy actually plotted against peak intensity. So in a way, with the peak intensity, you can have a bit of measure of how much of the proton of that particular compound, or if there are more compounds resonating there, that are actually sharing in that particular peak by measuring the intensity. So this is just a simulated spectrum, which you have already learned about the deshielding, the deshielding effect. When your atom, that particular nuclei you are dealing with, it's not surrounded by electronegative atoms like oxygen, like halogens, maybe it's not in the benzene ring, you will find them close to the TMS, pointing towards your right. So you call those upfield, they are shielded. So they are surrounded with a lot of electrons. But when the same atom, when you now bring it close to maybe oxygen, nitrogen, as the case may be, or it's even in a ring system like benzene, that system that has already been described for us. So we experience the shielding is moving downfield, and your value, you obtain it away from the TMS, higher value. So we call this area downfield. So you already have a background to this. This will be a little bit of a repeat also. So I won't be stressing here, but you can see when your hydrogen, which is colored in each instance here, depending on the environment, you can see the approximate delta value. That is the value for your chemical shift. You see the increase as the environment changes, getting to be close to your electronegative atom, depending on where the hydrogen is joined to. You have the values increasing. So it won't be a surprise that when you now have a double bond system, you have around 5 to 6. That is why if you have a terpene, for instance, a triterpene, a common cytosterol, there is a double bond around position number 5 in that molecule, and coincidentally, okay, it's between 5 and 6 if you are numbering your carbon. Coincidentally, that is the approximate data value. So you will also find your hydrogen in that environment to be coming around 5.1, 5.2, 5.3. So, and you can see on the right, the system where the hydrogen is even directly, attached to the electronegative atom. This time around, is not attaching to the carbon next to the electronegative atom. As you will see, take for instance, in the case of methyl ketone here. So it's not, the hydrogen here is not joined with the oxygen directly, but when your hydrogen is directly, then you cause more trouble, you cause more delay, and all, and you see the value coming around 9, let's say for this case 10 to 12. Okay, so what I would just like to say here, when you are accounting for hydrogens that are next to oxygen, like in alcohols, deuterated chloroform may not, in all cases, I think in most cases, it won't give you that peak, but DMSO, dimethyl sulfoxide, can give you that peak very, very well. So you might not find that peak when you are dealing with chloroform, although not in all cases. There are some peculiarities that you might still find it. So if you have aldehyde in your system, you will find it between 9 and 10. And most of the time when we are processing our NMR data for metabolomics, we still consider values up to 10. Reason being that after 10, you hardly find peaks, and it will be you will be interfering too much with your data because you have a lot of zeros after 10, except for special occasions where you have that hydrogen directly attached to the acid. So these ones, you can account for your maybe fatty acid, some other type of acid with whatever is joining to the carbon. So this oxygen, you already prove your compound with whatever this R is being is telling us, but this can easily be ignored, as this is just the only, as this case here, the only hydrogen type that you will find beyond 10. So it can easily be ignored. However, if you still want to look out for it, you will still see it in your raw data. Obviously, they will be there. Whenever you are now processing for maybe metabolomics, then you can actually exclude that type of peak and just work with between 0 and 10. So some applications, I will just be stressing a few things here. You can do qualitative. What do I have there? This is actually the most which I will still be stressing. What do I have? What type of compounds are there in my extract? But if you want to go deep into quantitative, you want to ask yourself the question, how much do I have? So there are protocols that can still be employed to get your quantitative NMR. You can spike your extract with a compound of known weight, and then but with a peak that you are sure will not overlap with the peaks already available in your extract. So if you are able to trace that peak out and is not overlapping, you are sure that that peak belongs to the compound you have spiked your sample with. Easily we can do the integration of the peak under the signal. When you integrate, then we can use that integration to reference the others, and you will easily get relative to that peak. So if there are only 1 hydrogen, 2 hydrogens, so relative to that peak, you will know how many hydrogens are below the other peaks coming in your NMR spectrum, and that can guide you on how much of the compound is there, or beyond that compound. Perhaps there are more compounds actually having overlapped NMR chemical shift. So it's a little bit of technical part of it, but we will not be exploring it as such for now. So let's focus on the qualitative identification. How do I annotate? You have already seen those charts, the table, and Prof has mentioned some of those guidelines that will, for a basic maybe researcher, you don't even have deep knowledge in chemistry. When you see a peak coming around 5 point something, you can guess, oh, maybe I have a double bond here, maybe I have an OH somewhere, an ether. So these are some of the class of organic compounds that you will find in your extract, the alkanes, the alkenes, the alkynes, arenes. Arenes are basically benzene rings that are just fused together. All of them, they are all hydrocarbons. When you begin to introduce hydrogen and nitrogen for oxygen, you can have the aldehyde group, you can have the ketones, you can have the alcohols, you can have ethers, esters, carboxylic acids, amine and amides. For your proteins or amino acid is just two groups combined together. You will see the amine group carrying the nitrogen. You have NH2 group or NH group. If it is quaternary, you have N group that is joined to any other thing, but it's joined to a carboxylic acid group. You will see COOH somewhere, you will see NH2 somewhere. So when they are together with a just like ROCONH2 or ROCONH something. So for that COONH2 coming together to form that junction in your molecule, you see they are amino acids. So with NMR, you will easily discover that, oh, on this compound, I have a carboxylic acid, I have an amine group, and then you can tell that I have an amino acid there. So generally, NMR is used in structural determination of primary and secondary metabolites, whether they are known, whether they are unknown, including isomers. There was a question on isomers. I will be showing you something just very shortly to throw more light into that. So when you use other techniques, take for instance GCMS or LCMS, you stand a chance of mixing up your metabolite, especially when you zoom into the specifics. Okay, this compound here, they say it's a flavonoid, but you will agree with me, for people with the experience, that there are cases whereby 3, 4, 5 flavonoids of the same group class, they will have similar fragmentation patterns. In some cases, you might actually find it difficult to establish the difference in their fragmentations. It can really be close. So in that scenario, you need NMR to now crack the problem. Okay, do I have a catechin, or maybe this other isomer of catechin that they have the same molecular mass? You can't judge by the mass value, the maybe high resolution mass, or the mass that you are getting. It's also hard for you to judge by the fragmentation pattern. Then you need to zoom in into the chemical environment. How are the hydrogens joined together? What is the slight difference between this particular flavonoid and this other one? Maybe it's just a rearrangement of the hydrogens, of a few of the oxygens inside the molecule. So with that, with NMR, you can easily do that. And we will still talk a little bit about the 2D NMR. I will be showing you very shortly. So you employ NMR. It's not limited to the highlight here. It's very robust. This NMR, you can use it in drug discovery. Okay, here we mean pharmacologically active metabolite. So if you are dealing with agricultural products or just a medicinal plant, what is your interest? Do you want to develop, get a drug, or you want to ascertain the authenticity of this crude? Okay, the traditionalists, they use this particular plant to treat cancer, to treat that other one. When you're writing your papers, for some of you, you must have hit that rock, and the reviewers come back, well, it's good, but can you give us an idea of the compounds responsible for these activities? So with my training, we isolate the compounds to the best of our ability. That has a lot of limitations also. So you isolate the compound. Sometimes you even do like single crystal XRD, X-ray diffraction, where you see the 2D picture of your molecule. Most of the compound you see that we have been displaying, they are just like flat molecules, but in reality, those molecules are not like that. There is a way they are oriented in space. most of those, and that is why the isomerism come into play. Most of the times, those other techniques cannot give you that type of details to tell us how is this molecule oriented in space, which carbon is joined to which hydrogen, is the nitrogen at the end of the molecule or is somewhere in between the molecule. So NMR will definitely tell us. So you can tell and give some quality control of your traditional, let's say, medicinal plant, and you can tell the reviewers, okay, these activities, I can trace it to this compound, that you yourself you will obviously test those individual compounds. You test it along the extract. So you test your isolated compounds, you test it along the extract. And then you can see when it's in form of a matrix in the presence of other metabolites, is it doing better? Or is it doing better as a single agent? There is this thing we call botanical drug. So botanical drugs, generally we deal with extracts, and because as soon as you start to separate the molecules, the compounds, the active ingredients, it's as though you are just killing that drug. And we have seen that severally, and the only way to explain that is even to bring in metabolomics into play, because for a natural product chemist, I will be isolating compounds in most cases that are of good abundance in that extract. There are tiny little compounds that I may not be able to get. The LCMS will pick it up. The likes of these metabolomic NMR-based metabolomics, we pick such trace elements, compounds out, and when you are able to pick that from your metabolomics, you can go back to the bench, you can go back to your LCMS and develop a method to now isolate, even though it's in a little quantity. You can now develop a method like preparative HPLC, high performance liquid chromatography. You do a preparative method and then you isolate that compound. And then you take it back to NMR. Is this compound, is the structure what I propose and all of that? So you can go down to food authentication. Okay, for people that are, maybe you have started your project and all of that, you can actually develop a database. You can have some people are working on, let's say, potatoes. You can develop a kind of NMR database that will serve a purpose in the future, whereby you have authenticated this particular variety of potato from that other variety, and if it is planted in this area, maybe in KwaZulu-Natal, comparing it with the one planted in, let's say, Gauteng here, you can actually have a database of the NMR, such that if another person is planting the same potato, the same variety in another province, then you can compare the NMR. Are there slight differences? Is there a particular difference in the primary or the secondary metabolites when you move across the provinces? So you can easily, and when there is adulteration, maybe you got to the market and you felt that, no, I doubt this product. You can bring your food back to the laboratory, buy it from the market, do your NMR. You must still get relatively the same profile. If they say this food was harvested from this area and maybe quality has been compromised and all of that, you bring the sample to the laboratory, run your NMR, overlay it, you superimpose it, or you stack it with your established database. You yourself developed this. Then you can tell, oh, I can see a peak at 4.3 that was not there when I did this experiment. So indeed, you can.
I think so.
Can we just ask you guys to mute, please?
Okay, can you still hear me?
Yes, I can.
Okay, thank you very much. Can I move on?
Yes, please do.
Okay, good. So you can bring that. It's also applicable to cosmetics when you harvest your plants and then you want to apply it in cosmeceuticals. You also want to test active ingredients in that particular extract. The best thing in drug development or even in cosmetics is to get what is active. You don't want to put the entire plant. So when you don't have the identity of the active ingredients that are there working on the melanin and some other maybe enzymes from your plant material, is going to be difficult for you to put your foot on ground and say that, oh, this particular product is helpful in cosmeceuticals because of this particular active ingredients. But a more advanced application is in medicine. And people have taken NMR further now. They are doing in vivo magnetic resonance imaging. So you can actually do NMR for what is happening inside your body. And some of us, we are familiar with magnetic resonance imaging, and it's still the same application this time around, just that it's dealing with in vivo studies. So it's not only on cells that you isolate, you put on ground. Then the method has been developed to really crack a lot of challenges in medicine. Now, let me quickly go to this slide. As you can see, you are looking at the anomeric protons of glucose. Glucose is a common metabolite, a primary metabolite. But I want to also emphasize here that you can't talk about secondary metabolites without making reference to their primary metabolites. The primary formed the secondary. And when your plant is being exposed to certain conditions or chemical conditions, some stress, the primary metabolites starts to modify, to convert to secondary metabolites, all because the plants also want to adapt to the environment and things like that. That is not the scope for today, but the primary are very important. As you can see, your beta glucose has your OH group there that is pointing like up in the structure, but on the alpha glucose is pointing down. And there is a proton that we refer to the anomeric proton. This proton here that from left and right is directly joined to oxygen. In a way, it increases the chemical shift because of the environment of those two oxygens. And you can see here the alpha glucose, we have his own anomeric proton, more deshielded to the downfield. But for the beta glucose, NMR will tell you when you are finding your anomeric proton there, then you know that you are likely going to be having your beta glucose. For people that have been doing NMR-based metabolomics, you will also notice that sometimes you see this long peak around maybe 4 point something, 5.2, 4.6. If you have been finding that, you can actually trace it to your glucose. But here we are, you can differentiate your alpha and beta glucose with that with the picture there. That is your HDO. Is deuterium oxide there that was used to run the sample. Okay, I'm going to pick this. So some of the applications I'll be showing now, most of them, if not all, are actually extracted or were extracted from some of our past work. So let me go to the next slide first. So in this case, we worked on bulbi natalensis. For people that are familiar with this plant, I'm not too sure. There is a market product. It's a testosterone booster. Athletes, they use it. It's in the market. It's no longer a product that maybe is being tested or so. It's in the market, and I've seen a lot of articles being published on the reproductive maybe effect of the plant on some on reproduction and all of that, how it improves fertility, and a lot of work has been done on that. But you will notice that from the articles that are available, we don't have a full profile of the chemical constitution. Some guys have been able to publish very nice articles on the plants, on the compounds responsible that may be responsible. Sometimes when you test those compounds, they have very limited activity. But when you test the crude, you discover that the crude has better activity, as the case is with bulbar nathalensis product. So it's the crude that is being used. So it will be good to have a broader idea of the compounds responsible, and that is why we have just about 19 compounds here. I did this with GCMS. But when this work went through the reviewer, they wanted us to have some more validity to some of the compounds. Okay, I won't be going deep into that. But I came back, then I did the NMR of the same extract. So the A, the one on top is I extracted that same leaves of bulbar nathalensis with deuterated, chloroform there. Ran the sample with deuterated chloroform. And for the one below, we ran it with methanol. And obviously, there are peaks. You see the deuterated chloroform. The chloroform extract there. The one on top couldn't pick much of the phenolics, but down here, because I'm using methanol, I was able to pick a lot of phenolics. Okay, just for sake of clarity, the plant material for the A was extracted with dichloromethane. That is DCM. Yeah, that will take a lot of non-polar compounds somewhere in between, but not purely non-polar, because not hexane or diethyl ether. Here I use methanol to extract. So after extraction, we just dry the sample, we air dry them, then we subjected them to NMR, but then using the available solvent, deuterated solvent here, which is deuterated chloroform, and in this case, deuterated methanol. So this is a way to annotate. And as you can see, you will see a lot of, let's say, 2 is here, peak for 2. Peak for 2 is also here. There is another peak for 2. You will see peak for 1 for compound 1. You might still see it elsewhere. So the idea is that I went into, also I did some isolation and I was able to get some of the compounds even pure. So went to the NMR of each of the compounds that were picked from GCMS, and then start to look at their NMR, trace where the peaks are located. And I was able to annotate 1 to 19 compounds, and there is the list. But more importantly is this structure. I didn't put all the structures there, but some of them are here. This is a triterpene, campesterol. This campesterol is close to cytosterol. It's even close to, so there are a lot of these sterols that we have similar structures like this. So the issue is that the position of this methyl group might not be here, maybe it's here. Maybe this, so that just a little bit of rearrangement there can create difference in the compound. However, when you do GCMS or LCMS, don't be surprised because they have the same mass. You might be getting the same type of profile for this type of compound. But when you isolate, when you zoom in into the NMR, you can easily tell whether I'm having this particular compound or the other. So you have this double bond here, you have the OH, you have this methyl at the end of the structure. This, okay, for people that are familiar with it. So when this is representing a carbon there with 3 hydrogens joined. So when you have this type of system, what you have learned in your basics of LMR, what Prof Prinsloo have already explained to you, you can begin to put them together. This methyl will be very low, maybe 1 point or 0.9, 1.0 thereabout, because it's not joining to anything. When you come to the methyl here, it might be higher. The reason being that now it's close to a double bond. Okay, when you leave the CH3, when you come here, there is a CH2 there, hydrogen, 2 hydrogens joined to a carbon there. There is a closeness to oxygen. And at this particular point, there is another hydrogen is not showing for now. That hydrogen is directly joined to the carbon link to the oxygen. So that is how you will elucidate the structure, because this hydrogen here will be different from the one on this double bond. This double bond, you will be getting it around 5 point something. All of this will be different from the one that is not even joining to anything. So that is how we play around with the NMR, and you are able to elucidate the structure. So those are the other molecules obtained from that particular extract. Let me just move forward. Okay, in this particular project, we have a collaborator that is working on some animals, rats, and all of that. And they try to do some anti-inflammatory studies on this extract. But they found out that the terpene-rich extract was able to effect some significant anti-inflammatory activity or property. And what I did, I got the extract. They did some methods to get the terpene-rich part of the extract. So you can say this is like a semi-purified, not really purified, but they took other things out. They focused more on the terpenes. So I was going to interpret this data from LCMS. We got the compounds, fine and good. But when our compounds go out, we still get some challenges. Okay, tell us more. How are you so sure that this compound is there? And I just did a quick work with NMR, and I saw that really you have your terpenes very rich in this particular extract. As you can see, the cycle on your far right. And the oxygenated region, obviously we have some oxygenated because even the triterpenes, they have oxygen. Some of them have some double bond. They may not be many, but you have a lot of CH2, 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 CH3, those other environments that are not close to double bond or oxygen or a benzene. In fact, the aromatic region is most interesting. In the aromatic region, I have zoomed it there. It is very, very low. So they have taken a lot of flavonoids out. A lot of flavonoids. If it's rich in flavonoids, you will see a lot of peak here, strong. So that particular extract actually had very good activities with the triterpenes. Okay, there might be some traces, but you can see very, very low. So with your Mestre Nova, you can do this manipulation. You zoom it in, and then you have that expanded region. Then you can still zoom into this region. And even if we go further, we can now pick each of the compounds and trace their peaks on this particular NMR spectrum. So that is what the extent you can go on NMR. I'm moving to the next slide. Okay, I won't be dwelling much on this. In this particular scenario, we isolated that compound. It's a coumaroy derivative. Dodesa, the name is there, trans-P coumarate. So we isolated it from this extract. Okay, fortunately, we took the same extract and we tried to do some green synthesis with the extract, and we found, okay, it was able to synthesize some nanoparticles used in some biological studies and all of that. We needed to know what compound was actually responsible. Yes, it's in the midst of many other compounds, but which one was perhaps the strongest? So I started stacking the compounds that I've isolated from this extract. There were about 10 of the compounds. There were flavonoids, there were alkaloids, and then I found out this compound had a very strong match, as you can see from the left. Okay, my further study on this discovered that instead of an ester bridge here, I had a low, long chain, maybe fatty chain there. Initially it was long, but when you come to the extract, that fatty chain, this aliphatic tail has reduced drastically. So from 2D, NMR, and some other analysis, I was able to find out that my molecule was originally like this, with a glucose. This group in the middle, a lot of peak strong there, is sugar region, as you can see in the middle part. I wouldn't know. Can you see my mouse moving around?
Yes, I can.
Oh, okay, so you have your sugar region there, and I was able to link it up with that same molecule. That no, this time around it's no longer a fatty acid ester, a coumarate, but the coumarin group is still there. This left side of the molecule is still there, but I actually add a glucose, like a sugar moiety attached. So what do we do when you say you are isolating compounds and things like that? Sometimes you tend to compromise some of the compounds. Sometimes in the course of even doing this and that, putting it on the column, doing your conventional chromatography, the molecule can modify a little bit. You can have some things dropping away. But when you take your sample raw as it is, you inject it into the instruments, it can give you a relatively good profile, fingerprint of the natural compounds that are there without modifying anything. Even though from maybe if I did some other type of extraction for this particular study, I would have got this aliphatic tail. You can't do green synthesis with maybe hexane, or so it has to be water extract. So this was a water extract. If I can recall vividly, maybe with methanol or thereabouts. So, but I ran the sample in methanol. Originally, the sample on top was run in deuterated chloroform. So these are a lot of things you can actually bring out from your NMR spectrum when you are dealing with it. Okay, very quickly, I'm going to take you through the interface with other equipment. As you can see there, I put the picture there just to emphasize the fact that our work is very complementary. You can't continue to do LCMS in isolation without making reference to NMR at some point. Yes, you publish a lot of papers, but there will be some limitations in the type of results and findings and your conclusions that you are drawing from your work. So we need ourselves. And that is actually extracted. I've tried to put the reference there. It's what's taken from a paper. You can see the reference on the left. But when you compare NMR with others, you will see reproducibility, things that you have been mentioning before. It's high here. The resolution may be low, but you can actually quantify. And then you have all other parameters to compare these methods. This is HPLCMS. This is still your LCMS. UHPLCMS, ultra high performance, and the details are there, but just a general comparison. So for this one, I won't be taking too long. GCMS is very sensitive. However, it can also be very, very confusing, especially when you run your GCMS and you depend absolutely on the retention time. That is a big error. I only found out not too long, unfortunately for me. Reviewers threw my work back to me saying that no, you must use retention index, not retention time, because the databases that actually the NIST and other databases, they just take into consideration the retention time, and they try to compare on this database, what do we have in this retention time? And if they see a little bit of similarity, perhaps in the fragmentation pattern, they give you your suggestion. So your GCMS, be careful not to rely too strongly on your retention time. You play around with your retention index, and then you can have a good, valid result. But if you come to NMR, NMR will give you better validity on the identity of the metabolite that your GCMS is giving to you. Okay, as useful as, okay, this was just a graphical abstract from that article there. So compound ID, when you really want to crack the compound identity, try to go for NMR. You can see the tilt is not too much, but that is when we are talking about compound identity and reproducibility. Your LCMS can either be HPLC, we have already talked about that. I move forward. So to the NMR techniques. 2-dimension. Okay, Prof has highlighted a few things about this 2-dimension. Okay, it's there on the screen. I'm trying not to spend too much time here. Your COSI, all of this I've in my research, I've used COSI a lot. Okay, I leave that homonuclear. When you come to heteronuclear, I've also used HSQC and HMBC a lot. Then when you go through space, I've used NO-C. I've not used RO-C, depending on what is available to you. But they do similar thing. HSQC is a little bit different from HMBC. I will just show you maybe a few spectra just now to highlight more on these two-dimension NMR techniques. Okay, when you see this molecule, this is the COSI spectrum of rutile-carpine. Okay, this was one of the alkaloids I isolated much earlier during my PhD. Okay, I didn't number the peaks. I will number, the number is there in the next slide. Let me quickly show you. So if you check the numbering here, you will see that 7 and 8, they are there on these two peaks here. And on my compound, that is 7 and 8. 7 and 8. So I go back. So now to, sorry. Okay. Yeah, so for this COSI, this is 7 and 8. COSI will tell you which hydrogen on which carbon is joined to which. So in this case, if you draw a line, you can see that you can correspond these two peaks. So if you draw a line, diagonal line there, so it's saying that 7 and 8, the position 7 and 8 hydrogen, they are next to each other. They are next to each other. There is a carbon there, there is a carbon here, but both of them carry hydrogens, and it is this NMR, this 2D, that is telling us that really the hydrogen of 7 is next to the hydrogen of 8. Can you see how it separated them from the benzene, the aromatic systems? So that is the only system where we don't have the double bond close by, or the benzene close by, and it separated them very well. So when you zoom in into this region, you will now see by drawing perpendicular line there, you will know which hydrogen is joined to which one. So you will always get 9 is joined to 10, 1 is joined to 2, 2 is joined to 3, 3 is joined to 4. So that is the correlation that 2D will give to you. Very, very practical. It's real. But just doing ordinary proton NMR or a carbon-13 NMR, they won't give you this type of information. Okay, in this scenario, this is now HSQC. That is another type of 2D NMR. You will, in this case, the correlation is from the proton to the carbon. The proton to the carbon. So on my y-axis, you have the carbon. On my x-axis, I hope I'm not mixing that up. That is fine. I think I'm correct. Okay. So I have the proton. So if you check proton number 7, it's here. Proton number 7 is there. It's joined to carbon number 7 with this correlation. So you draw a line there, you draw it there. So it's joining to 7. So you have 7 carbon, and then you have 7 proton. The proton is around 4 point something, as you can see here. If not up to 5, yeah, around 4 point something. Then the carbon is around 40 something, maybe 39 or 40. You can see. So carbon we give you that. But when you get to 8, proton of 8 is coming around 3 point something. So, but the carbon of 8, it's very low compared to the carbon of 7. So it's around maybe 20 or thereabouts. It's there. So you draw that line, you get it. So that is what 2D can give you. You get the exact identity of your molecule. Okay, if you recall that we did mention earlier that nitrogen 15 is one of the nuclei that we also perform NMR on. I was going to confirm whether my compound is real or not, the alkaloid, whether it's real or not. Then I did nitrogen 15 HSQC. Nitrogen 15 is supposed to tell me the hydrogen that is linked to a nitrogen. The hydrogen that is linked to a nitrogen. So it's one of the ways, even though there are other ways, you have the mass spec and all of that. But the NMR tells me that in this molecule, only one nitrogen is joined to hydrogen, which is the one here at position 13. Only one nitrogen is joined to hydrogen because it's a 2D. So you get a nitrogen peak, you also get the hydrogen peak. So you do the correlation, say, oh, now you are happy. This one has no hydrogen, you don't bother. This one has no hydrogen, you don't bother. So you don't care about those ones in this type of 2D. You want to correlate the nitrogen to the hydrogen. And that is a perfect example for your nitrogen 15 NMR, which you use to confirm alkaloids, as the case may be, if they carry the hydrogen. Other things that would be needed here is the mass spec. The mass spec, if you do high resolution mass, as I already told you, you can't do NMR and say, no, I don't need the mass. No, we need ourselves. So the mass spec will tell us how many carbon, how many nitrogen. It will give you that this compound has 3 nitrogens. You will see N3 as part of your molecular formula that your high resolution mass spec will give to you. So just to mention, we have not reached this level, but it's already existing. You have literatures, you have papers already being published from hyphenated NMR techniques. So this is a very, this is a good way to go in the future with this Metabolomics society or group. Let's reach a level whereby we can hyphenate. So you don't have a trouble. You run your GC, it's already hyphenated with NMR. So at the molecule, the compound is coming out, the NMR detects that particular compound. You can say it's because you have let's say for your chromatography, you have different retention times, and you hope that one compound is supposed to come from this particular retention time. So as the compound comes out, elutes in that at that retention time, the NMR picks it up, then you quickly have a profile, then you can confirm, oh, really? It's that compound, and you have your mass, obviously, because it's hyphenated technique. So I'm not going to be going into details. This is a very much more advanced hyphenated techniques. This is LC, UV, SPE, NMR, MS. Okay, I'm not sure how many institutions has this. So the LC chromatograph separates. You have your chromatograph, you have the retention time. It goes through a UV, you have the UV spectrum. We also elucidate structure using UV. Then it still passes through the solid phase extraction again. And then before now coming to NMR, NMR give you the chemical shift of the compound coming out, and then the mass spec now picks it up, give you the molecular mass, give you an idea of the fragmentation patterns, and you move on. What a very robust study there. Okay, there was a question on pH. Let me give you another practical illustration of how pH can affect your study or your work. This was also part of the project that I did during my PhD. So I was isolating from this plant called Umlungumabele in I think that should be in the Zulu language or so. So it's Xanthocylum capense, Xanthocylum capense. So I struggled with this NMR spectrum for almost a year. Okay, I isolated the compound. I never knew there were 2 compounds there. I never knew there were 2 compounds. You kept on isolating. I was getting some fragments of the compound, but it never came together. Okay, not until I did the mass. When I ran the mass, they were able to get 2 distinct masses. Oh, then I quickly tapped my head. Yo, so I had 2. Okay, thank God for that breakthrough. So I now came back. Okay, how can I annotate one from another? I will not be showing the spectrum for now, but I was able to annotate these 2 compounds distinctly. Each of them came out neatly from the spectrum. Okay, this is where I'm going. What happened? I couldn't separate them. Okay, I also had limitations, the type of chromatography we employ and all of that. So there were also limitations. So I couldn't separate this from this, but I didn't know why. So while trying to write up this compound, it's a non-compound, not a novel compound as at this time. This other one, also not a novel compound. They are very wonderful alkaloids. You can see the nitrogen there. So they are very amazing alkaloids with good activities. So as I was trying to put my work together, it was between 2014 and 2015, only for me to stumble on this paper. So as I was struggling with the right part, you can see my right side there. These guys were sitting together, sitting down to understand what is happening between these two molecules. So they can actually interconvert. They interconvert at maybe some changes in the pH. So this figure, I've taken it from that article there. Okay, this is my own taken from that article, but reference 38 that you see here is actually pointing to this paper. Okay, I was almost going to be the first one to do this discovery, to have the discovery and all of that. But you see, when you are doing your research, please don't sit with it. Publish your paper because somebody is out there doing something similar. I had to reference them. So these were the first guys to link up these two molecules. And then they discover that the first one, it's at the pH of 6.4, but the second one will be happy at the pH of 9.2. You can see, so the details are there. For people that are much interested, you can get the details, but in my case, because they have very close properties and all of that, I couldn't separate them. The NMR were very, very related, but I just had to reference that. So you can see what pH can do in your samples. Okay, just to round off, these are additional notes. Just what I picked from the discussion so far, your prepared tip. If you are using a clean wrap, you stand a chance of introducing plastics. If you have been doing a lot of GCMS, there is this compound that always say diactive phthalates. These phthalates always come with maybe GCMS results, and sometimes you even think, and people have reported phthalates. Unfortunately, your phthalate is actually not part of your compound in most cases. It's coming from the tip or from the clean wrap or from anything that is maybe plastic related, because that compound is what they use as plasticizers in most of these things that we use. And then versus organic solvent. So how do I mean here? Your polar organic solvent will not dissolve that compound easily. So we have problems when we are using maybe dichloromethane and then, or maybe chloroform. But I still want to thank people that have discussed already, and Prof giving some light. Okay, don't stay too long with a pipette tip. Make sure you change it. But then there will be leaching, as the other professor has mentioned. If there is a leaching, if there is a trace of plastic in your sample and you have already done your experiment, it's not the end of the work. So when you are about to go into chemometrics, when you're about to convert your data to your spectra, to data that your software can pick up and do your plotting for you, the peak can actually be excluded. So what I do on my software, some other NMR processing software that I have, I overlay my spectra with diactive phthalates. If I can see match, then I know there is probably some trace of that molecule, and then I can trace it out. I can ignore them, I can exclude them, and then it will not interfere with your results, or so you account for it. So you can easily account for it if your work has gone that route, or you have done a lot of GC and you have seen those phthalates a lot. So make sure you avoid them to the best of your ability. But in a case they are there, you can still win by just taking notes, overlay your spectrum on the spectrum of diactive phthalates, and then you can remove it from it. Okay, automation and manual. NMR automation and manual. The instrument we currently have in CSIR, I am aware there is another instrument that is actually automated. Maybe we need to explore the possibility of, okay, the instrument we have can be automated, but to the best of my knowledge, for now it's being run manually. That can really be demanding. We really want us to explore the possibility of migrating from a manually run NMR to automation. Automation, you just need to prepare your samples. Maybe I think the machine can take up to 50. For the one I was using during my PhD, I think it takes up to 50. So you won't even introduce any error because your 50 samples are queued all at once. The machine gets there, picks it, runs it, picks it, runs it until the 50 is completed. Then you can do the next 50, just depending on how much sample you have. That will be what we really need to consider. Okay, we have talked much about this. So how big your magnet will tell you how resolved your NMR chemical shift will be looking like. So you see a lot of overlap sometimes. It just looks like a noise. If it is giving you a noise, but it's there, maybe with 400, you try 600, you will discover that that noise is not really a noise. It's not just anything. There are 4 peaks there, or maybe 8 distinct peaks. So those are some of the things that happens when you use a very low strength NMR. But with higher NMR, you can get your results quicker. You can use less quantity of samples, and then you have a better resolved NMR spectrum. Okay, this will interest a number of listeners. If the chemistry have been too much for you, the chemometrics will not be too much. When you are able to now extract information from chemical systems by data-driven means, we say you are doing chemometrics. So what happens, I will not be going into detail because that is what will be covered tomorrow. So what happens is that spectrum you are looking at, those things that just look like noise going up and down, your softwares will convert them to data, to figures, 1 point something. So all your data will no longer be like a picture. It will now be taking the form of a data, a numeric data. So with that type of data, now you can do your clustering, your plotting, and which will actually help you to solve a lot of problems. You can do comparative studies. So I'm just going to read this. It says, chemometrics is inherently interdisciplinary. So we can collaborate any type of project using methods frequently employed in core data analytic disciplines, such as multivariate statistics, applied mathematics, computer science, in order to address problems in chemistry, problems in biochemistry, problems in medicine, even in biology and in chemical engineering. So I will be stopping there. I want to believe you have been able to pick 1 or 2 things from the discussion. Thank you.
Thank you so much for that, Dr. Budere. Let me just, okay. There seems to be an echo again. Okay, so we had a question. I'm just going to go up to the first one. I'm just going to ask our other participants to just mute their mics. So we'll just have our speaker unmute. I'll just pose the question. Okay, there was a question on the interaction of the proton with, I think, thiols. How does the proton or your hydrogen react in the case of thiols?
Thiols. Okay. Okay. Something that has a sulfur, is that what? Just to get it right. If the person can maybe throw more light to that. Thiols is THIOLS. Am I correct?
Yes. So yeah.
Okay. So in this case, you still your protons will still resonate. However, we have not explored NMR for sulfur, but when you run your sample, the sulfur will be behaving the same way oxygen will be behaving, like a similar way. So it's a kind of analog for aqueous. You will also know that aqueous can easily be converted to thiols. In fact, they are derived, thiols are derived from aqueous. So the sulfur will simply behave as oxygen, maybe with just slight difference. So you will find the hydrogen also deshielded because of the effect of the sulfur. So similar effect that oxygen has, your sulfur will have.
Okay, thank you for that, Dr. Budere. So I think if you remember your periodic table, you'll see that sulfur is in the same group as your oxygen. Let me just check. I don't think. Okay, William has a question. What are the major differences between the instrument megahertz? Maybe I missed it in the talks or presentation. In terms of resolution, is higher megahertz instrument always better? And would that include having to conduct less runs for any sample type?
Okay, so what I've noticed is that, in natural products, sometimes you isolate very small milligrams of sample, and if your instrument is 400, you will struggle to see all the peaks, especially, let's say you isolate 5 milligram sample. With my experience with a 400 instrument, I always load a sample that sometimes can be up to 10 milligrams, 15 milligrams. But if I'm coming low as 5 milligrams or lower, some of the peaks will not be, you may not even see the peaks. You might see something tiny. So you will be confusing yourself with the experiment because you would think there is no compound. However, the compound is there, but the resolution, it's really, really low. The signal is very, very low. So as you move higher, so we take that same sample, if it is 5 milligrams, if I take it to 600, I get a better result. But if you are fortunate, you now take it to 1,000 or 800, you will see all the peaks. Everything will come out and it will be distinct. And Prof, if you remember, Prof was talking about multiplicity spin splitting. So if you have a peak, take for instance a doublet. A doublet will appear like two signals coming together, but they will stand beside themselves. So for a doublet, when your NMR is not well resolved, there is a tendency that you will not see it as a doublet. You will see one broad singlet. It's a bit broad, but it's now getting a little bit of split on top of the peak. Something will tell you, is it a singlet or is it a doublet? Maybe it's because you are using a low resolution. When you put it on a bigger machine, the two peaks, it's one signal, but it's split to two. Those two appearances will be very, very distinct. The most confusing is the triplet. You see, in a triplet, you have one peak very long. You have two other peaks that are actually coming beside that one peak. So when you are using a low resolution or when you put your extract, you won't see those three peaks distinct. Sometimes you just see a shoulder. We call it a shoulder. So there'll be a shoulder on the left, there'll be a shoulder on the right, and then there'll be a long peak. Actually, if you have used a stronger instrument, a bigger magnet machine like 800, 1000, you won't see that shoulder. You will rather see a distinct 1, 2, 3 appearances of those of that particular signal. So it's one signal, but it's a triplet. It's appearing 3. So that is the difference between your machines, 400, 600, and the likes.
Yes, Dr. Budere, can I just ask you? I think your screen is sharing.
Okay. Remind me again.
You can stop share by the cross to the next to the mic. To the right. Yeah.
Okay, stop sharing.
Yes. Yeah, so what I've done also is I know of a paper where I think a 400 megahertz and a 600 megahertz instrument was compared. I actually posted it on the chat group for those of you who are interested. So definitely higher megahertz definitely is better resolution and I think sensitivity as well.
Yeah. Okay, yeah, I'll also check that.
But of course, some institutions only have a 400, and so it might be better to have a more concentrated sample or more of your sample then.
Yeah, correct. That's correct.
Okay. Are there any other questions? How does the profile of active, let me just see, I can't see the full. How does the profile of active molecules, meaning phytochemicals for a sample, tell of its medicinal values, for example, antimicrobial properties?
Okay.
So how does the active molecules tell you a little bit about the medicinal value of a plant?
Okay, so the first point of reference will be to individually look into the compounds, each of the compounds, and quickly do a brief of literature search. I think there might also be some databases that probably has maybe or maybe some materials where you can have a list of antimicrobial compounds. So that there have been books that have been published on that, maybe a few articles that, okay, over this period of time, we have highlighted all the active antimicrobial compounds from this class of maybe this particular species or these genus or even this family. So you have that that pool of compounds. So you get your compounds, you check it individually. If it falls into that category, then you already know that you have an antimicrobial compound. If it has not met a database individually, there will be, that is, if it's a known compound. If it's a known compound, you can just do your literature search, a quick search. If you can play around with search criteria very well, you can easily know whether they have done antimicrobial studies on this compound or not. So when you get to the paper, also make sure you read carefully to know to what extent was this compound active. Because some articles, they just report the activity. But really, if you go to the MIC, you go to the IC50, you just notice that, no, it was just tested. It was never active in the first place. So that will be a good way to go. I think Prof Prinsloo has also done some work, even with this metabolomics. By the time maybe it can be of help with that. They have done something similar to that with anti-HIV. So they pulled some plants together, and they were able, with metabolomics, they were able to figure out, okay, these plants, we see that these ones have antimicrobial, antiviral activities. And they were able to trace out the compounds. They were able to pull the compounds responsible for the activity. Okay, then they now went further to find out, okay, which one is common for this particular set of plants. So there is a way that metabolomics can also assist us in pulling that activity out of our medicinal plants and the likes. Yeah.
Okay, thank you for that answer, Dr. Budede. I'm not sure if Prof Prinsloo wanted to add anything.
No, I think Dr. Budede answered quite well. So yes, first would be to, if you have the name of the compound, then it would be easy just to go to literature, find if that is there. If you're not sure about the activity, you can still do a search on, for instance, similar compounds or the group of compounds, especially in the species. And yeah, I think I'll maybe just also include that example later on to show if you have plants with similar activity, then you can get common compounds from there, and then which are probably then the active ones from your samples. And then, yeah, then take it again to literature and see what's available, and if you can link it to activity.
Okay. And I would think that a good backup to this would also be to do obviously bioassays then, just to confirm as well. Just normal biochemical assays in the lab to confirm the metabolomics findings.
Yeah, if it's not available.
Yeah.
As Dr. Berere mentioned, most of these compounds normally are already tested.
Available.
So, yeah. And also, but it's also good to link your findings to your original extract. So if you do assays on your original extract and you find antibacterial activity, then it's easy to link it then to your compounds if you have compound names. Then it's like the puzzle is fitting.
Definitely. I don't see any other questions. Is there anybody with a question? You can raise your hand and unmute. Okay, so with that then, since there's no more questions for this session, I'd like to thank Dr. Budede then for showing us the applications of NMR, specifically how you applied it in your research. You mentioned there the magnetic resonance imaging that is also sort of used to get metabolic data. And in that sense, when it is used where you actually derive metabolic data also, they usually refer to it as metabolic magnetic resonance spectroscopy. So with that, obviously, a lot of people are familiar with MRI. So it's similar to, going for an MRI. But when they're doing the scan, there's an additional sequence that is worked in that actually records metabolic data. So that's where the in vivo aspect comes in. The thing with magnetic resonance spectroscopy, even though you're getting in vivo data, is that the peaks are less and obviously less resolved. So NMR sort of increases the resolution of your peaks compared to MRS. So I just wanted to add on that. So thank you for showing us those applications. I see Tayo has a hand up. Please unmute. You're free to ask your question.
Good morning, everyone.
Hi, Tayo.
I'm Taiwo from Nigeria, and I would like to ask one question, but I don't know if this is going to be out of the box, but it's something I'm interested in, and I think I can bring it to the floor here to get a direction for it. So, the presenter made mention of chemometrics, which is something that caught my attention regarding the kind of project I've been doing for a while. I have been looking at evolution of sperm morphology across species of birds. And just with this presentation, I got a little bit interested in some metabolomics kind of research where I can look at ejaculates and see what's what's possible across ejaculates, across species, and what is there that I can really look at across species when I convert these, those spectral we talk about, and do kind of analysis looking across species, because that's also part of whether sperms that have high competition, we also look at sperm competition in this case, and baby sperm that have low competition, how do they vary in terms of this, molecules that you look at when you're looking at metabolomics. So we can do some comparative studies at this level and see what can be the findings that we'll get out of it, which can be informative in explaining the differences in sperm competition that we can observe across species or maybe within species, depending on the situation. So this is my thinking just while he was presenting this kind of presentation. So I don't know how possible it is to do this kind of work, and what suggestions with your expertise in this field can you provide for me to expand this area of research? I don't know if I was clear with what I presented as a question.
Tao, I think I just missed. So what sample type are you looking at?
Well, sperm cells.
Stem cells.
Yes.
Sperm cells or stem
birds.
cells?
Yes. What?
So did you say stem cells or sperm cells?
Sperm cells.
From birds?
Yes.
I'm not, okay, I'm sure it is doable. I'm sure the sample that you're talking about sounds more or less similar to, your serums or your plasmas or your
Yes.
CSF, right?
Yeah.
So in that case, it would mainly just be an analysis where you get rid of your protein. So we would basically filter that sample to get rid of proteins and then subject that to NMR. So it shouldn't be difficult to do at all. I don't know, Shane, if you can maybe comment.
Pardon?
Dr. Mason, if you've seen anything regarding that.
That is a whole new biofluid I have not looked at, so it'll be interesting to look at the literature behind that.
Mm-hmm.
I'm sure someone has touched on that before, but I have not, on my side, looked at anything like that.
Yeah, I've also not read anything related to that sample type, but in my mind, it should be handled more or less similar to what we do with CSF or serum or plasma. But if you are interested in starting something like that and maybe just doing a trial run, feel free to reach out to us. I'll post my email address as well, and Dr. Mason can leave his as well, because we do more of biofluid type of analysis, and we can see and maybe discuss and see whether we can work out a project from there and see if we can try out some preliminary analysis.
Okay, thank you very much.
Have I answered everything that you queried about? So you just wanted to know whether it is doable or whether such a sample type would be whether it would be possible to run it, right?
Yes.
Yeah.
And you did say it's possible, so which means something doable. So you can give a try to that.
Okay. Are there any other questions? Okay, so Prof Prinsloo, we have one more session, right? One more speaker, Dr. Nongemane. So I think we will take a little bit of a break. Is it okay? Hi, everybody. Are we back? I think we can just give a minute or so, Prof, and then we can start again.
Yes, sure. Dr. Nongemane will not join us today, but myself and Dr. Nongadotsi will just share some examples.
Okay. Just check, there's a bit of a noise coming from somewhere. Okay, it's gone. Okay, it is now 12:42, so I think we can start, Prof. I'm going to hand over to Prof Prinsloo just to elaborate a little bit more on examples of NMR-based metabolomics. Over to you, Prof. Prof Prinsloo? Okay, while we wait for Prof just to start up that presentation again, a reminder to all delegates, and perhaps maybe share the reminder with some of your colleagues and collaborators. For those of you who have signed up for tomorrow as well, so we've sent individual links for the respective days. So there's a specific link for the 9th of September. So please make sure that you click on that specific link. We will also post a feedback survey. We ask that as many of you please complete that as possible, because we do actually then use that to sort of better or improve on our workshops. It's also important just to show to our stakeholders, people who assist us, you know, whether it's helping you guys or not, and what the specific need is out there. Okay, Prof Prinsloo, are you back? Okay. Are there any questions from you guys in the meantime? Okay, if not, some of the aspects also, so when I started earlier today, I just recapped on a few of the concepts that was mentioned yesterday. Because I attended another meeting, I just reviewed a little bit of what was done while I was away. So I think Dr. Lufuna also covered a little bit about, you know, the experimental layout. What's the order, you know, planning your experiment, designing it, then moving on to the actual sample prep, putting that sample through the machine, getting data, pre-processing that data, and working then to actually interpret that data. So part of what you mentioned was the aspect of multivariate analysis. So multivariate analysis just basically looks at the simultaneous analysis of more than one variable. So you're basically looking at everything at once, because if you can imagine, a lot of our systems are complex. The interactions are complex. So it's very rare that one single variable or compound or metabolite will explain the interactions that are happening. And so you look at it holistically in a multivariate sense to sort of get an indication of that complexity. So you look at everything. So you look at all of these variables simultaneously that might influence your system. The principal component analysis, or PCA, that was mentioned, just to remind you that that shows the natural separation of your sample. So you can get a lot of information regarding your samples. So if they are very dispersed or very close, you can then comment on how homogeneous your particular sample in a test group is or was. And then there was also the aspect of PLSDAs or OPLSDAs. And she mentioned a little bit about removal of outliers as well. So just keep in mind that the removal of your outliers is guided by biology. And you can also use your statistics to sort of guide the removal of outliers. So just remember that the removal of specific samples or groups is not random. So it should be guided by your biology or sort of controls within your statistical model. And of course, as I've mentioned to you guys earlier, we will have a symposium later this year in October, where some of the research across South Africa will be showcased, specifically metabolomics, obviously the metabolomics research. And there we are looking at specifically adding a section on, doing certain quality checks on your data as well as validating your models. So just keep that in mind, that we will then have the symposium later this year in October, and that such a session will then be added on. Prof Prinsloo, are you ready for us?
Yes, I'm ready.
All right, I'll hand over to you.
Okay. Okay, yeah, so what we'll do is we're just going to show a few examples of work that's been done to give you an idea of the applicability of this and how it can work and what you can do. So I will show some examples of work that we've done. Unfortunately, these are only plant samples, but I think you'll get the idea of what you can do and what answers you can answer with these technologies. So I'm going to do a untargeted example as well as a targeted one, and then one where we played a little bit with the information. And then after that, Lefuna will, Dr. Namadotsi will also show you one on the soil metabolomics that she's done, so you can get an idea of what's happening, what you can do on soils as well. So this project was where we collected quite a number of plants, all with antiviral activity, and we want to see if there are similar compounds in plants that confer the activity. So it's all plants from different species, different genera. So it's not plants at all related. It's unrelated plants. So we just want to see how we can make this, how applicable this is to something like that. And what you see on the screen is that this is a OPL-SDA plot of the plants, and then we group them according to the activity in literature reported. So, for instance, the yellow group is plants that's active against one type of virus, and the green one is another type of virus. So you can see this was amazing to see that this, based on the chemistry and the activity of the plants and a specific for a specific virus, was grouped together. So and then we decided to focus on one of the groups, which is the light blue one, because that was quite a strong group, and that was plants with anti-HSV, herpes simplex virus activity. To see if we focus on this, can we see what are the common compounds of these plants? So these are just the kind of things you can do. So this is a 3D plot. You can see there's the light blue group, the group with the anti-HSV activity, and they are the rest. You can still see there that there are definitely groupings within that based on the type of activity of the samples. And the hierarchical cluster analysis is also a good analysis to do, to just verify what you see, and because it's working in a completely different method. And even there you can see the light blue group was separated from the rest, the red separate, the green, yellow and the green separate. So this confirmed the results that we got in the previous, in the OPL-SDA, which then tells us something about the model, and that we are not overfitting, or that we are not looking at data that is not valid or that can't be interpreted. So if you use methods like this, some validation method, and it helps to make sure that what you see is true and that it's worthwhile continuing with your data. There are other methods as well. I will talk a bit about some of them later on. Then we normally construct a contribution plot like this. So the bars at the top is showing the positive association So these are your NMR regions. We'll look at this tomorrow at how you get to this, but these are your NMR regions. So this is your ppm, so it's from 0 up to 10. And these are then the NMR regions that are then positively associated, in this case then with that blue group, the group with anti-HSV, herpes simplex virus activity. And then if you start to look at these areas that was strongly associated with that, then we start to match that with compounds. And from these, we then got to a compound. This is chlorogenic acids, but there are a number of chlorogenic acids. And these peaks were all aligned to the chlorogenic acids. So by using this metabolomics, we used then unrelated plants from all over the country, some of them not even in our country. And by looking at similar activity of the plant, we could then identify the areas of importance that is similar between those plants, and then we could derive at the compounds that are common between those plants. And if you look at this, the chlorogenic acids are well known for their antiviral activity. So so that worked well, and that gave us a good answer on the study. So this was published in Metabolomics. So if you want to get a bit of more detail on this publication or on the work, I just highlighted a few things. Then you can check the article. So that was untargeted. So we just looked at a few plants, see what we get, see if we can get some common compounds from there. So it was just an untargeted analysis. So in this study, this was on moringa. We did a targeted analysis. There's a lot of information available on moringa and active compounds identified in this plant. So in this case, we looked at harvesting frequency, which is also an interesting application of metabolomics, to see if your chemical profile can stay the same, if there's variation in your profile, if you harvest more often or less. And that gives you an idea of how you must harvest or prepare your samples for commercial production of products from moringa. So in this case, on the left, that is a PCA. So you start with your PCA just to get an overview of the samples and of the variation in your samples. You can see the high frequency one all clustered or moved to this side, where the intermediate and the low harvesting was more or less to this side. Some that was in between, but it was clear that your high frequency samples definitely moved away from the rest. So when we do a PLS, an OPLS-DA, which just means we add some information, we tell the model, add this information to this data as well, and then you analyze it. So in this case, we just did then a group of high frequency and low frequency, and then you can see we got much better separation, except these ones, but that was something different. We later on saw that these samples were, 2 months of very high rainfall, so that influenced the samples. But in general, the low and intermediate frequency harvesting separated from the high frequency. And here we did some validation. So this we call a permutation test. And this is just where you use your model, you take out some or the program, you use the functionality where the program takes out some of your values and replace it with random values. And if you then replace it with random values, your model should be not as strong as previously, or not be as good as the previous ones. And this is what you find here. The green is your original model, and the blue is the permutated one. And then the difference in here tells you that the model was not as good as the previous one. and there are some factors that you look at here, but this then support the model that you are working with. So this is one validation method that you can use. And then we had a look at targeted metabolites. As I mentioned, in moringa, there are many compounds that are known and that we are known that they contribute to the activity. And this is a region from 1 to 3, and this is a region from 6 to 8. So these are your phenolics, your aromatics. And the red peaks or the red profile is the low harvesting, and then the blue one is the high frequency harvesting. And then you can see that your high frequency is then lowering your compounds, most of them, not all of them, but many of them, as your NMR is quantitative. So a lower peak means there's a lower concentration of those hydrogens. So in this case, most of our phenolics, of our flavonoids, then increased in low harvesting, and in high harvesting it was decreased quite a bit, which then is very important if these are your medicinal compounds, because if it decreased this much, it means your material will not be as active as it should be. So in this case, in the region from 1 to 3, here we looked specifically at, this is GABA, and you can also see the GABA was increased tremendously when it was low harvesting when compared to your high harvesting. But what I just want to show you here is, for instance, there are peaks of a compound that were not affected by the high harvesting frequency. So it's not as if all compounds are increased or all compounds are decreased. You get this variation. It depends on the metabolic pathways. It depends on the type of compounds. Some primary metabolites are differently affected by things like harvesting and your secondary metabolites. So so here you can clearly see that some of your compounds still stayed high with a high frequency. So it's not as if a low frequency harvesting frequency was resulted in an increase of all our compounds. So, but as I mentioned, this is now targeted. So this was GABA, and these compounds are mostly things like mostly things like caffeic acid, ferulic acid. I think there was also chlorogenic acid in here. And most of these are well known for their medicinal activity. So then we could use exactly the same analysis, and we could then quantify it as well. So we quantified 4 compounds: chlorogenic acid, ferulic, phenolic, and GABA. And you can there clearly see how the compounds increased with the low harvesting, and it decreased with the high harvesting. And yeah, this is the value of NMR in terms of quantification. Of course, you use the exact same analysis that you started off with. And as this quantitative method, you use that and you can quite accurately determine the concentration of your compounds in the sample. So once again, if you want to read a bit more about this, I just highlight a few things of the paper, but this was published, and so you can check the paper if you're interested in more details on this. The last example I'm going to give to you is, I'm just going to discuss, is one where we looked at, for instance, seasonal variation. So obviously, if you harvest material for a product or for use, then you do not want your material to differ in your quality between seasons. This is also important. Plants are seasonal, so you can't harvest right throughout the year. So it's important to have information on what seasons you can harvest and which not. So on the left, we have, for instance, the 4 seasons. So this was winter and autumn and spring and summer. So you can see already here the 4 seasons separated. out, which tells us that the chemical profile differed quite a bit between the different seasons. And what we've done here then is we just took the ones with the most difference to the ones with the least difference. So we compared the spring with the winter. So we just did a pairwise comparison. So in this case, we had 4 groups. But in this case, we just wanted to see if we compare 2 seasons, what are the main differences between the 2 seasons. So we removed some of the data and we did a pairwise comparison between spring and winter. And this allows you then to drill down a bit better and also to see your differences between your samples more clearly than in the case like where you have 4 groups. Once again, we did a permutation test with 100 permutations to make sure that the model is valid. And once again, you can also see the green ones are the true values. The blue ones here are the ones that was replaced. And you can see the difference between the 2 models, showing that the original model was much better than what happened afterwards when you replaced some of the values. Once again, we did a contribution plot. So these again on the axis here as your NMR regions, and the ones at the top was positively associated with spring. What we've done here, I'm not showing it here, but we've done the biological activity here, the antibacterial assays. So the ones in spring was much more active than the ones in winter. So so these ones here were then the ones that was associated with a higher activity, and these were the ones that we concentrated on. From this, using the databases, things like the human metabolome database, and then from there we saw it's not the compound that we originally thought it was. So then we had to go to literature on similar type compounds to see if the peaks can match with other compounds that are very similar. And then we derived this compound, which was never previously isolated from this plant. So, and this is the difference, I think, when you use LC and NMR. With LC, you rely a lot on the compounds that were previously isolated. As for instance, with the isomers, you cannot really identify them. So, so in this case, this was a similar compound than others that was previously identified, but and when we check the literature on this is also linked to the questions earlier. When we checked the literature on this, we saw that there is quite a bit of information in literature about this compound being active against bacteria, although it's never been isolated from our plant. And this compound then matched the peaks here, which means that in the season where we have high activity, this compound was then in a higher abundance, and then probably then resulted in the difference between the 2 seasons because of the higher abundance of this compound. We have not isolated this, so this is the next step that should probably be happening now, is to isolate this compound and confirm that this is really one of the major contributors to the activity of this plant. So also to make sure that we are targeting the right compound, we then did LC-MS. Once again, as mentioned previously, you have to use different techniques to make sure that what you have is correct. And by using the LC-MS, we were then searching for this compound. And indeed, this compound was present. And we could also see on the LC-MS analysis that the abundance of this compound was much more in the active seasons than in the non-active seasons. So confirming or then using 2 different analytical techniques to make sure that your conclusions and the interpretation of your data is correct by not making wrong conclusions from your data. So once again, I've just highlighted a few of the things of this work. So if you're interested in this, you can go and read up a bit more on this. So, but these give you a good example of combining NMR and LCMS to get your compounds. So so that is all that I'm going to show here. I think maybe Lufuno, maybe you can maybe also give your example, present your example, and then maybe we can do the questions maybe at the end if there are questions on
Okay.
all of them. Yes.
Okay, Prof. Can I go ahead?
Yes, please.
Okay. Prof, can you assist me? I want it to be on the presentation mode. I'm getting confused.
I think it is in presentation mode. Lufuno.
Here it is.
Oh,
Yeah.
okay, that's fine. All right. Okay, good afternoon, colleagues, and our important MSc students as well as PhD candidates. I'm going to be giving you a talk on some of the results from my PhD study. I'm saying some because it's just a portion of what we did. So for this particular talk, the title is going to be Metabolomic Analysis and Microbial Composition of Soil Supporting Bouquet Africana Growth. I think I need to state that this study was conducted at Department of Agriculture and Animal Health here at UNISA, and as well as Department of Biochemistry in the Wageningen University in the Netherlands. Okay, so allow me to give you a brief on the background of the title. Bouquet Africana tree is a leguminous tree, as shown on the top right. These are the pods that it bears, as shown on the top right of the picture. It's a leguminous tree that is that grows in the savanna woodlands in non-calcareous sandy soil. And in South Africa, it's mostly found in provinces such as Mpumalanga, Limpopo, Gauteng, and Northwest. This tree grows in clusters, so you find them in groups. You can never find them like individually. They're always in groups of trees. And the pores that are as indicated are only bear the pores around January and July. But this does not happen on a yearly basis. Some years it may bear trees of pores, and some years it may not. We still do not know. I think that's open for another research that we need to do perhaps. So this tree is known for its medicinal properties. Traditional healers all over Africa have used it to cure several ailments or diseases such as your sexual transmitted diseases, for an example, gonorrhea and syphilis, inflammation. It has been used to cure the back. I'm talking about the back now. It has been used to cure inflammation, malaria, inflammation and dizziness and headaches, whereas the roots of this tree is used for treating poisons and snake bites. Now, above all, this tree is also a host for Serenafoda caterpillar. I don't know whether the picture is clear enough. This Serenafoda caterpillar uses this tree as the only source of its feed. And I think I also need to indicate that the Serena caterpillars are regarded as a delicacy among the Bavenda, Batsonga, and Bapedi tribe, who are found. These tribes are found in Limpopo provinces. Maybe let me just clarify something. So, Serena is not the same as Mopani worms. What we know is Gonimbrasia bellina Westwood. The difference is that Serena only use Bouquet africana as a host and feeds on Bouquet africana leaves, whereas the Mopani worms use this Colophospermum tree, or what we call it Mopani tree, as its host, and serves and eat only feeds on the leaves of this Mopani tree. So sorry, I'm just, there's something that just came up of the screen. That's why I'm a bit confused. So the two are not the same. However, they serve the same purpose, and they also share the same common name, commonly known as mashonja. So you can just imagine that the sentimental value that this study had on me as someone who comes from Limpopo and a Muvenda person. So that is why I think I fell in love with the study. And as indicated, the Serena is a delicacy among my people, or people from the province from where I come from. And this is just a meal. Sorry, this is just sorry, I don't know what happened. Yeah, so this is just a picture of a meal of a well-cooked mashonja that is going to be eaten with a hard porridge or pap. That's how we call it in South Africa, pap or hard porridge. Now, between the two, which is Serena and the Mopani worms, or the Gonimbrasia bellina, the difference beside the host, the tree as a host, is that Serena is yellow. It has a yellow and black cuticle, whereas the inside is yellow and it has a salty taste, so it's mostly preferred. Whereas the Mopani worms has the cuticle. Mopani worms cuticle is black with white and a bit of reddish on the back, and the inside is black, and it leaves a bitter taste on your mouth after you've eaten. So those who come from the province knows what I'm talking about. The mostly preferred one is the Serena So in as much as there are so many benefits about the medicinal benefits and the Serena that we consume, that comes with this tree, this bouquet of Africana trees, farmers were having headaches and sleepless nights because none of them could transplant or propagate this tree out of its natural habitat either through pods or excavation of seedlings. So they were having a problem, and that is why this tree is not found anywhere else. Like, you cannot find it in the nursery, and it is also not commercialized. And so that is why we had to come in now as scientists to say, what is it that we can do to find out exactly what the cause of the problem? Why is it that whenever the farmers are growing it outside its natural habitat, it will grow, but with stunted growth. The growth will be stunted, and then 6 months later, this tree will die. I also need to indicate that the study was people are coming in. The study was conducted at Telperon Game Reserve, and the choice of this Telperon Game Reserve is because you find a lot of clusters. You find this bouquet Africana trees in clusters. Like, when I say clusters, I mean like thousands, hundreds of bouquet Africana trees at once. So that is why. And it was also some kind of an interest to the owners of the game reserve. They also wanted to find out that what the cause was for this bouquet Africana not to be difficult, what was causing difficulties when it comes to propagating this tree outside its natural habitat. Okay, so the first thing that we had to do was to go to the basics. And when I say basics, I mean we had to go to the field to Telporen Game Reserve. And I want you guys to have a picture of these two pictures somewhere, just save it somewhere in your head, because it's gonna come handy as I go along. So we had to go to the game reserve, and as you can see, the picture here on the bottom left, I'm gonna call this bottom left, there are no trees there. They're just small grasses and rocks. And then whereas the one on top right here, it's trees. These are the clusters that I was talking about. So between these two, it's just what separates these two is just a single lane of gravel road, single lane. I think it's even not even single. I don't think it's single, but maybe 800 something. But it's just that's what separates these two. So you come here, you find, as you are driving or as you are walking, you will see on your right or on the left, there will be Bouquet Africana trees, clusters of them, lots and lots of them. And then you look across, just a look, without even stressing your eyes. It's empty. There is nothing. And so this is the picture that I want you to have in mind. So we had to go there, and then we had to collect the soils. So I also need to also bring you to the new word that we come up with. We had to come up with words like non-bouquet soil and bouquet soil. So bouquet soil indicates the soil that was taken, soil samples that were taken or collected where the Bouquet Africana trees grows. And then the non-bouquet soil means that the soil were taken where there were no Bouquet Africana trees at all. So this was taken, and the soil samples were collected and taken to ARC, Institute of Soil and Soil, Climate and Water in Pretoria, so that they can run or the test for pH, for your total nitrogen, phosphorus, potassium, your nitrogen, your nitrate, and ammonium, as well as calcium. We also had to find out about the level of organic matter or the percent of organic matter in those soils. I think I either downloaded a wrong, yeah, I think I did. Sorry about that. I'm sorry, can I just reshare? Can I be given the permission to share again?
Yeah, Ilfina, you can just
I think
stop and just.
I downloaded the, yeah, okay. I think I downloaded the wrong, okay. Okay, stop presenting. Mm. 5. Okay, I'm gonna set this as final.
Prof Prinsloo, while she sorts that out, there's a question on harvesting.
Okay, let me just check here.
When working with plant tissue, how important is it to separate the main veins on the leaves from the remainder of the leaf?
Okay, this is the first time I got something like this. I'm not sure if it will really make a difference. As long as you harvest the same in all your samples, unless there's a specific reason why you do not want to have the veins in there. But for me, the most important thing will be there to make sure that if you include the main veins or not, to just keep it the same in all your samples, because maybe there's a reason for this, for you to not want to have the main vein in there. But yeah, just make sure that all your samples are treated the same. But I don't think in general the main vein will make a difference.
Thank you. Dr. Lufuno, are you ready?
Yes, I'm ready. I'm hoping that the screen that I'm looking for would pop out.
I guess if you don't have it, you can just talk on it and then.
I don't have it. Yeah, I don't know. I think the devil is trying me. Anyway, so yeah, so we had to do the basics, and the basics was to, as I indicated, we had to check the physical and chemical composition of the soil, of both soils, right? And the only difference that popped out was, or that the findings was that it was only nitrate and ammonium that was higher on Buckia soil as compared to non-Buckia soil, and so that was not enough for us to say that was the main reason to say that's the difference. So that was not enough. So we had to now move into metabolic profile, and I'm not going to bother you with all these because we did this yesterday, and Professor Prinsloo, there was a video that Professor Prinsloo showed you guys on the extraction methods all the way to NMR. I think the only thing that I need to brag here now is that we do not have 400 megahertz. We have got 600. So yeah. So and then of course, so today, this morning, you saw how Mestronova is done. After you've gotten your raw NMR data, then you need to now export it to Mestronova so that you can do the pre-processing, the normalization, the base and phase correction or baseline correction, as well as the alignment of peaks, standardization, and so on. So once we did the pre-processing of our raw data, we now had to bring in the statistical analysis, and I indicated yesterday that we do not use one-way ANOVA and two-way ANOVA here. We use a multivariate statistical analysis with the 3 or 2 different preferred models that we use. So we started with PCA, which is the unsupervised model, and there was really nothing that we saw, and that is why there are 2 or 3. So once you start with one and it's not really giving you something that you can report about, then you need to now employ the next statistical model. And in this case, we included we had to employ the OPLSDA, which is the supervised model. And as you can see, the green lights, the green circles are the non-Buckia soil, whereas the blue circles are representing the Buckia soils. So you can see that there is a clear and distinct separation between the 2 soils, which told us that there is indeed a difference in the metabolites, or in the compounds, metabolic compounds metabolic compounds that are found in both soil. So this for us was an aha moment. It was like an aha moment indeed, because we had something to work with now. Okay, so after this, after realizing that yes, there is a difference between the 2, of course, statistically, we had to now annotate. We call it annotation of compounds and using genomics. But this here that you are seeing here is a contribution plot. And the contribution plot will show the NMR regions, NMR regions. And so the ones that are on above the x-axis, these ones here, your choline and the betaine and the trial, these are the positive, they were showing the positive regimes on NMR. What do I mean? These are the ones that were found on Bukia soils. These three compounds, or the 4 compounds, were found on Bukia soils. And then when we check on them, comparing to non-Bukia soil, there was nothing. I mean, not nothing. I mean, these compounds were not found. The compounds that were found on Bukia soils were not found on non-Bukia soils, and vice versa. So Professor Prinsloo mentioned something in the morning. I think somebody asked, why do we have to use NMR if it's less sensitive than LCMS and GCMS? The answer, he gave you the answer. He said, it is because NMR has the ability to also pick up the sugars. Now, had we used, we also used LCMS on this study and GCMS, Prof. We used the 2, we used the 3. Now, LCMS only picked up the amino acids, 22 amino acids, but it did not pick up all these compounds that we find that you see here. So NMR for us was a breakthrough. It was the says it all, if I have to put it that way. So as you can see, the regions, the sugars, especially with the trehalose was dominant or predominant on Bukia soils, showing from regions of 3.6, 3.6, 3.8, 3.9, and 5.2. Betaine was on 3.3 and 3.9. That is betaine. Then we had 2 compounds. I'm sure you're wondering, what do you mean by choline and carnitine-like? So these are the compounds that they when we were doing the annotations, they look exactly like choline, but they were not choline. They also look exactly like carnitine, but they were not carnitine. I'm going to tell you why I'm saying they were not. So, and all both these 2 compounds, the choline and the carnitine, they were found on the regions, NMR regions of 3.1, the 2 of them. So we only gave them carnitine-like, and I think that's the beauty of doing PhD, you know, it teaches you to be innovative and you think out of the box, and so all these compounds that I'm talking about here, the one that showed the positive correlation on Bukia soils, were identified using spiking methods, you know, where we use a reference standard, we buy these compounds, and then we spike the process, or the extraction method is the same as when we started, like the one that Professor Prinsloo showed you yesterday. The only difference with identification is that you are going to use a reference here for identification. So if it's 100%, then you know that's what you got. And I'm not going to bother you. I decided not to bore you guys with the chemical structure of trehalose and betaine, but these 3 were found on Bukia soils. Now, on the non-Bukia soils, we found acetate. We also found acetate was on 1.9, formate was on 8.4, and lactate was on 4.2, I think, if I'm not mistaken, Prof. So these 3 were found on non-Bukia soil and were not found in Bukia soils. Now, literature review will tell you that acetate, lactate, and formate, most of them are in growth inhibiting compounds. They are growth inhibiting compounds, which means that is the reason why Bukia africana was not growing just across the road, but were growing on this side. Now, the trehalose and betaine, we had to name them as growth promoting metabolites, because these were the metabolites that were actually influencing, or the influencing, and yes, influencing. English is just second left, but these were the compounds that were that played a huge role in the growth of Bukia africana trees, and that's why we call them growth promoting metabolites. Now, with that said, this was not enough. Again, we had now to look at, we had found the answers, yes, but we wanted to find more. And more meant in this instant meant we wanted to comprehend the microbial diversity of the soil communities that was happening in the soil, or the soil microbes that was happening. We wanted to find out how diverse, how the similarities and all of that. And this was done with the use of metagenomics. So soil DNA was extracted from both soils. When I say both soils, I'm referring to Bukia and non-Bukia soils. So it was taken, we collected the soils, and then they were taken to Ingava, biotechnology. That's where, okay, extraction was done at UNISA for us to find out if there is, we're going to find any strain of DNA, and yes, it was found. Once we found that, we send the soil for sequencing and identification at Ingava biotechnology. And this picture that you're looking at now shows the this is a representative of fungal species in Bukia soils. I decided not to bring in the kingdom and classification of kingdom and the families and all of that. I just wanted to come straight to this, because this is the says it all again, you know. So as you can see here, Penicillium, which is a fungal species, showed predominance. We can see pictures.
Dr. Buder, yes, someone says they can't see the pictures.
Can you see them, Prof? No?
I can see them.
Okay.
Perhaps just refresh your page, Judith.
Okay. Can I do that?
I can see yours, Dr. Budere. Judith, if you refresh, maybe just log out and log back in. Can the other participants see the pictures?
Yes, I can also see.
Okay.
Yes, I can see it too.
I can see.
Okay, so I think Judith, maybe just refresh. She says she can see now. Dr. Budere, I'm just going to remind you that it is 1:34 already. So we've slotted, I think, the session until 1:30, right? Yeah. So I'm just going to remind you a little bit about time.
Okay. Dr. Namados, thank you. Okay, so, yeah. So Penicillium showed a high predominant, and it was the main discriminant in the Bukia soils, as you can see there. And then on non-Bukia soils, somebody's coming in and we're almost closing. On non-Bukia soil, there was a lot of uncultured and unidentified uncultured fungi that could not be identified. So that was, for us, was the main discriminant, that in Bukia soil we had a lot of, or high prevalence of Penicillium fungi, and whereas in the non-Bukia soil there was uncultured and unidentified fungal species. Okay, so in conclusion, we can safely say that the growth of Bukia africana tree is it's influenced by so many factors, such as soil metabolites and soil microbes, because there is a variation. We saw that there is a variation in the soil metabolites that were found both in Bukia versus non-Bukia soil, and there was also a variation in the soil composition between the two soils. So that on its own will tell you, or will tell us that, told us that the supremacy, the metabolite supremacy and microbe supremacy plays a role in the growth of Bukia africanus tree. So once you excavate, or once you try to grow it outside, you are actually disturbing the growth promoting metabolites as well as the microbes. That is why it dies after 6 months or so. Okay, so the ultimate goal of the study, what came out of this study? Of course, she graduated, and the title changed. And we also published a paper on metabolites, and this was, for us, it was great. It was a great feeling to publish at a high impact factor journal. It's 4.9, but Prof, I always say 5 because you can always round it to 5. So yeah, with that, I would like to put a few people that I want to acknowledge. I don't know if back again, but I would like to acknowledge the Oppenheimers, and the Oppenheimers are the ones who funded my study, NRF as well, and UNISA, Bazari, so that, you know, and those funding actually helped with all the lab work that happened. Were expensive, but it was not a problem because with funding. I also want to thank Professor Prinsloo and Professor Vervoort. I mentioned about him yesterday, and so I would like to thank my brilliant supervisors, and I also like to thank Metabolomics South Africa for you know, teaming up with Professor Prinsloo in this workshop. Thank you.
Thank you so much for that. A lot of passion and enthusiasm there.
Yes, ma'am.
I'm going to ask, is there any questions? I didn't see any in the post, in the chat. Perhaps you just want to raise your hand and ask your question. Did you do targeted or shotgun sequencing?
Judith, are you referring to the metagenomics or NMR?
The sequencing work, the metagenomics.
We did. No, it was not targeting because we did not know what we were looking for.
Okay.
We didn't know what we were looking for, so we just said they must just do anything, and so so I guess that is
shotgun sequencing.
Shotgun sequencing, yes.
Any other questions? Okay, if there are no other questions, I'd like to say thank you to everybody for sticking it out. Yeah, it was a productive morning into the afternoon. So just to recap, so yesterday we touched a lot on just the sample prep and then actually loading that sample onto the NMR. Today we covered a lot of the principles and the applications of NMR, specifically looking at where it is applied in the different you know, fields in terms of your plants. And of course, tomorrow we will now move a step ahead. I think Prof has announced that there's certain software programs that needs to be downloaded ahead of time. Please make sure that you log into the Teams link that has been sent with the date labeled 9th of September. Please make sure that you log in on time, since I guess the idea is that Prof wants everybody to work alongside him as he's doing this analysis. So please download the software, make sure that you're on time and that you click the appropriate link. And then, of course, yes, we will go from the point of the sample has been loaded. What do you do with the raw data that comes from there? So I think it will be quite holistic in that we would have gone from sample prep to analyzing, to looking to analyzing that data and then looking at applications. So I see there are a few comments. Ovia just says, thank you very much. This was such a great, insightful and informative session. Pleasure. So if there's anything else, Prof, perhaps you want to say something regarding tomorrow?
Yes, I just see there's a hand to Beatrice.
Oh,
is that the
I missed
question?
That. Yes, please. Oh, sorry.
Please go ahead, Beatrice.
Yeah, sorry, good afternoon. I downloaded the one of the softwares, but the second one, I'm unable to download it. That's the Mestronova. Each time I try to install it, it will go back. It's not working. It's telling me maybe it's dangerous or something. So which particular website or company can I download it from? That's what I want to ask, please.
Okay.
Yeah, so it's sold by Mesrolab, but I'm sure you just check it. It's Mesrolab. That's the only company I know that sells this software.
Okay, that's the one I tried downloading from, but it's not installing. When you get to maybe the final stage, then it stopped installing.
Is it not perhaps linked to your institution, Beatrice? If you maybe try download it at home on your, I don't know if you're working, if you're using maybe a company laptop.
I shouldn't use the institutional internet, right?
Yeah, maybe
Oh,
just try a different network. I don't know.
Okay.
Prof, is there no trial version instead of
Yeah, you must just also make sure
to the trial version?
the trial version or the evaluation version.
Yes.
Yeah, that might
Prof,
also be part of it. Yeah.
Yeah, Prof Prinsloo, can we perhaps maybe just in the chat box just post those names again for the soft?
Yes. Yeah.
Okay, so and then yes, just make sure that you download ahead of time and that you are then logged in by 9 o'clock. Okay, so Prof has posted the names of the software that you need to download. Okay. Yeah, so my company does not allow downloads. Yeah, so I don't know if you have an alternative device, maybe. All right, so Prof Prinsloo, is there anything else from your side?
Yeah, so if you cannot download the software, it's okay. We will show stepwise tomorrow on screen what you do. I will also, there's also a number of documents that that I will send to you, maybe post in the chat as well, which also show the steps that you take. So if you just follow then tomorrow on screen and just keep the notes next to you. So if you have your own software then at the stage, then you can just follow the steps and at least you know what's been done. So we'll show everything on screen tomorrow.
Okay, and we'll post, you said you will post the notes, the software notes as well in the morning in the chat. Okay.
Yes.
All right.
Can I just say something, Prof, before we go? For those who are working on soil, either thinking of working on soil, please, you can read the article that was published on soil metabolomics, and so that you can start thinking about your study and what else can you do. It will come very handy.
Perfect. All right. Well, thank you all for staying tuned and for coming back, being disciplined. See you tomorrow at 9 then.
Good.
Thank you. Bye.
Take care. Bye-bye.